Okay, good evening, everyone. Thank you for joining us this evening here as we listen to William P. Davis, or as we all call him, Will. As Will present his Masters of Science in Physics presentation. Will, of course, is a graduate of Seton Hall University. He graduated 2 years ago, class of 2019. And then he decided to continue onward to not only get a bachelor's of science in physics, but get a master's of science in physics. And Will has been working over the last almost 2 years now on his master's thesis, which deals with using plasmas and interacting them with agriculture. As part of his the thesis committee, I'd like to recognize Dr. Alper Shahiner, who is our department chair. So Alper is here. So good afternoon, Alper, or good evening, Alper.
Thank you, Jose. Good evening, everybody. Thanks for coming for this such a nice day at 6 p.m. to listen to our beautiful talks on physics.
Yeah, it was 90 degrees in South Orange earlier today, so it was actually really hot. The second member of Will's thesis committee is the Reverend Dr. Father Jerry, as we call him, Gerald Father Bonapane, from the Department of Chemistry and Biochemistry. Father Bonapane, how are you?
Very well, Jose. Thanks for having me. Thank you.
Thank you, Father. And I am the third member of Will's thesis committee. If you don't know me, I am Jose Lopez. I am a professor of physics in the department, and I have been working with Will since, I guess, since sophomore year, right? So we're going on 4 years now, Will, together. Mm-hmm. So Will today will present a presentation entitled Investigation into the Role and Generation of Rns. He'll explain what that is. In atmospheric plasma jet for plasma-assisted agriculture. So our master's thesis candidate is William P. Davis. Will, please.
All right. So he explained the title just now. So I work in the lab of electrophysics and atmospheric plasmas down in the basement. And I'm going to give you somewhat of a teaching, more trying to make you understand what the study was about, what the focus was, and how you do the experimental process in terms of plasma research for atmospheric-based plasmas. So we talked about who the master's thesis advisors. I just want to say thank you to everyone who helped me in terms of that for my master's thesis and guiding me and being able to help out in terms of that. So what is the presentation going to be on? We talked about it. It's low temperature plasma, but specifically we start off with plasma. We focus on what the project is, low temperature plasma, and then the interactions with agriculture or bioorganic organisms. Then how does that interaction, like what is going on there? So a lot of research entails the RONS species when you look at plasma agriculture and just low temperature plasma in general. And then we're going to see what the study entails. So that was just the background information. So what the study entails, how the procedure went, the results discussion, and then a comparison between different papers that saw similar effects. So first off, it's going to be more of a discussion. I don't mind if people ask questions at the end, but this is to make people understand or just get some information from this presentation. You leave with something that makes sense to you. So plasma is defined as the fourth state of matter. I think many presentations have said that already. And that fourth state of matter in terms of energy of the matter, molecules of atoms. It's a quasi-neutral soup of charged particles with the properties of a conductor that are used in many different applications, and just remember it's an electrical conductor. Sometimes you can describe it with heat as well. So these are different examples you might know in terms of plasma. So the aurora borealis, lightning, a plasma ball, neon sign, arc welding. You might be familiar with all of them, but I'm sure some people know at least one or two. And then we focus on what are the applications of this plasma. So before you saw some natural phenomena in terms of plasma, now we're looking at the applications. So this illustration shows various fields of industry using plasma in some form as part of their processing procedure. We see our telecommunications, textiles, automobiles, optics. So almost every facet of life might have some type of plasma processing, or at least an industry will have some type of plasma processing in their procedure. So moving further, right, what are these specific applications in research, right? We just saw industry, now this is research. We see here pulse vapor deposition for solar cell development that uses a plasma nitriding for surface coating resistance, that's the use of a plasma, and then plasma gasification for more environmental studies, that's the use of a plasma as well. And so what are these plasmas being used and how are these differentiated in research by the effects? So they're differentiated by thermal and non-thermal. So in thermal plasmas, the particles are in thermodynamic equilibrium at about 10,000 Kelvin to about 1,000,000 Kelvin. And you see there with the illustration, it shows the electrons, neutrals, and protons are around the same temperature at about 100,000 Kelvin. I have an illustration with the sun. Most of it is plasma that's in thermal equilibrium. In non-thermal plasmas, the electrons contain much higher energy while the other particles can remain at very low temperatures. So you've probably already seen this before from other plasma applications, but it's just to illustrate that, yes, that's the reason you can touch it at room temperature, because the protons and neutrals are very close to room temperature while the electrons have most of the energy. It could be around 10,000 Kelvin. I don't know what it is in the electron volts, but they use Kelvin in terms of describing it. And so we just described it in terms of temperature. So now relating that temperature to the different examples we talked about previously, this graph illustrates the electron number density versus the temperature of different types of plasma phenomena seen in the universe. These parameters are of importance to describe the physical different properties of matter in a physical nature. And so you see here different types of plasma we just discussed. I talked about the sun. Spherical tokamak is for fusion. So we're more or less working in the low temperature, high density range for our plasmas. But these are some of the examples you might have seen before: ion propulsion, lightning, and dipping into the low temperature, high density region. So just remember, we're focusing on that. So what are low temperature plasmas? I just described them. It's when the electrons have most of the energy, and the physical parameters of importance that we just talked about as well, where the gas temperature, the electron density, because that's how they describe it in terms of the density of the molecules coming together, and then the specific electrical parameters used to excite it. So because of their unique parameters, low temperature plasma can produce unique chemistry at atmospheric conditions. So sometimes they can be generated in a vacuum chamber, such as for like thin film coatings, different other types of coatings as well, surface etching. But sometimes they're generated in atmosphere, which is the focus of this study, right? We're looking at a low temperature plasma jet that we generated in atmosphere. And so how are they generated in atmosphere? What was the study that undertakes, or the science behind how you were able to generate it in atmosphere? So the focus of that is specifically Paschen's curve, because it describes the relationship between the necessary breakdown voltage of certain gases and their pressure gap distance. So the pressure plus the gap distance between the electrodes of the gas flowing through the electrodes of a plasma generator. And this just illustrates different gases here in terms of it and what their breakdown voltage and their pressure plus gap distance may be. You can see here helium is the one of focus. It's very low in terms of the breakdown voltage necessary for it. So when you start off with that, further research into it provided an understanding that certain low percentage admixtures of the gas, when I say low percentage, more or less than 20%, would have an overall effect of lowering the overall breakdown voltage of the gas. So combining about 5% admixture to nitrogen gas will allow for applications in certain atmospheric plasmas with lower breakdown voltages than nitrogen gas would have normally. And that's specifically around the output around here where the plume is in terms of the plasma jet. Okay. And so when I said we added helium gas mixture as an example, we actually used helium gas in this setup, in this experiment. And helium gas gives that low breakdown voltage, or how it does is because the feed gas mixes with the APPJ and lowers that breakdown voltage of air. This is because helium is very small compared to the chemicals in air. And then an effect known as penning ionization allows helium to transfer energy to other molecules in the gas. And you'll see that relates back to how you create excited species in low temperature plasma. And this is just showing here the helium atom discussing the formation of a nitrogen molecule. So moving on, what is the penning ionization that we just discussed as one of the things that is able to transfer the energy? And we discuss it because it is of focus in creating these RONs, or reactive oxygen and nitrogen species. So penning ionization is when the helium metastable is able to transfer energy to molecules in the air, such as nitrogen and oxygen, and excite them into an ionic state more easily than from direct voltage breakdown. For this reason, the effect is noted as this is related to the chemical creation, like I just said, of RONS, which we'll see further in the presentation, and this makes the chemistry unique to the plasma in whatever gases you're using to generate it or the environment it's being produced in. And this just shows pending ionization between two excited helium atoms, and it creates ions and then ejections of electrons. So that's what you need to focus on when you see reactive oxygen and nitrogen species. So we just discussed atmospheric plasma, the effects of how it's generated and how we generate it in atmosphere and create the cold plasma itself. So this graph just illustrates the gas temperature and electron density, the two parameters we discussed, of various atmospheric pressure plasma sources. So you see some thermal plasmas, and these are the non-thermal plasmas. We're focusing on DBD and APPJ, helium-based DBD that can be converted into an APPJ system. Okay, so what is the DBD? Now we're discussing what the DBD is because this whole study is to characterize this jet and discuss what phenomena we're seeing. So DBD plasmas are generated by using a dielectric placed between two electrodes using an AC voltage. Our configuration uses a high voltage electrode, but no ground electrode in a cylindrical configuration similar to the bottom right. That's this configuration. You see a high voltage electrode, but no ground electrode. That's what our schematic is, and you're going to see it. I'm going to show the schematic for our setup. So before we even talk about that, one of the main focuses is how these reactive oxygen and nitrogen species get created. I discussed pending ionization as one of the mechanisms for reactive oxygen and nitrogen species, but this chart is from Professor David Graves. He's one of the papers I'm focusing on today to characterize its jet, and you'll see another one from Dr. Friedman. He's a big inside the plasma industry, and then someone else from India who does a lot of plasma research. But from Professor David Graves, this chart just illustrates the excitation mechanisms that you might have already known, such as ionization, dissociation, sometimes there's recombination, dissociative recombination, as I discussed, and pending ionization. So all those different effects we just discussed to create these different mechanisms leads to the creation of the plasma. So what can the plasma do? Well, the plasma can be used in agriculture, and some examples of this is plasma treatments of seeds, plasma activated water, and the plasma localized plant treatment. So this is actually our plant being treated right here. That's not the correct distance, but it's just a demonstration. So what is plasma agriculture? I just said it's plasma related effects, but that includes germination effects, plasma chemical generation, and plasma surface treatments, which can be for the creation or breakdown of chemicals and cells. So you see here's a couple examples. You don't have to focus on what's in the middle, but it could be increased germination yield, plasma generated nitrite fertilizer, pesticide degradation, many different things. And finally, we get to why are reactive oxygen and nitrogen species special in terms of plants and in terms of analysis, in terms of these plasma jets, because they're the main focus of what's being generated in the plasma jet. So from atmospheric plasma chemistry is the creation of reactive oxygen and nitrogen species, I said, from the plasma. Low temperature atmospheric plasma has the capabilities to create these numerous reactive oxygen and nitrogen species in different concentrations. And when you bubble the effluent, which is the gas coming off when the plasma is created, into water or onto surfaces for further research regarding analysis and interactions, you can find some very interesting research going on there. Why? Because RONS is a big study in biological and chemical research, not necessarily physics, but now you can see why it's related to physics as plasma is becoming more and more integrated into industry and different parts of industry. So RONS can be produced from the environment in many different ways and have different effects due to high and low concentrations within biological cells and tissues. However, plants and animals produce compounds known as antioxidants to combat the effects of these RONS, known as free radicals, known as the oxidation reaction. There's different names for it, so if you hear something like that, yes, those are all the same thing. And so in small concentrations, you'll see here the physiological roles. They can be good in terms of cell signaling, in terms of cell division, differentiation, a regulation of apoptosis. But in very large concentrations or very high concentrations, you have an overwhelming saturation of it. There's lipid peroxidation, cellular damage, nuclear acid damage. So it's just limiting the concentration in there. And as I said, when there is too much, how do plants react? How do cells react? They produce antioxidants to stop the reaction. But let's discuss what that even means. So that oxidation reaction, the reaction between the free radicals and whatever it is there, it's ripping apart the whatever molecule it is. that antioxidants combats the oxidation reaction by breaking the chain reaction within the initiation stage of the reaction. So this is the initiation stage. It'll be reacting somewhere here to terminate it, and this leads to the termination, and the antioxidant is used up as a result. So just keep in mind that these all have short lifetimes. More or less, the RONS have much shorter lifetimes than antioxidants based on if they're used up, but the RONS definitely have short lifetimes. When I say short lifetimes, within nanoseconds, picoseconds in terms of that. So basil oil is the focus. Basil plants were the focus of this research, and there's a reason behind that, because basil plants and their extracted essential oil have components that are known antioxidants, phenolic compounds such as eugenol, and linalool is not a phenolic compound, where they are, but they are contributing to the antioxidants, and they directly are used up as a result of the oxidation reaction, and their antioxidant activity can be identified and determined. So if you determine the concentration, you can do that, and you can determine antioxidant activity percentage. So this is a chemical formula of eugenol, chemical formula of linalool, and then some basil oil, and examples of the different colors, because you can have different concentration contributing to the colors of the basil oil. And so then how does this integrate into industry? How is this useful for people in society? What did you find from this? So basil oil can be used as a preservative, and there's many different preservatives already in use, synthetic ones such as BHT and BHA, and they're added to foods such as cereals and lipids, and they have been linked to different illnesses, so that other alternatives will have to be investigated now. So basically I'm saying is that the BHT and BHA have been linked to like lung cancer, and I can cite the papers in terms of that for different things in terms of the FDA research. And so different alternatives are now being researched, not necessarily only to basil oil, but other different oils. So this picture here gives an illustration from a different study that showed rosemary extract on the right, and then on the left, there's nothing on these untreated meat. And so you can see it turns grayer than the treated meat. So this prolongs the state of unantioxidants, or like not being oxidized, where this is oxidized and becomes more gray and gray. And so basil oil can be used as a preservative similar to the rosemary extract by prolonging the shelf life of different foods, by stopping the oxidation of foods based in lipids. So it's mostly oils in terms of that, but other foods are oxidized as well, and they go stale. We know that. However, this is more the main focus because the meat industry and a lot of other baked goods and shelf-stable goods are based on lipids as well. So that was the basis of what the study is, what the study focused on, why we focused on basil plants, why we focused on low-temperature plasma generation. So what are the goals of what we're trying to find? Right. So the first part is to just try to investigate and quantify the RONS species being produced from our specific plasma jet. So investigate and quantify the effect of the low temperature plasma treatment on basil and its oil. So the basil growing, so the growth rate, as well as the extracted oil. Determine if there's any relationship between the plasma reactive species and the treatment on the basil plants, and then characterize it based on literature. So I always think that now if you ever do a presentation, try to relate it back to a study you've seen in literature, because it's going to make more sense to people when they see it. Okay, so the first part of the characterization was physical characterization. We just looked at it with measurements using rulers, calipers, just to see what was inside of it. So this is just a guideline. I'll get to the actual characterization when we get to the results. Next was the electrical characterization. So we used different electrical probes and oscilloscopes to look at the parameters from 26 to 30 kilohertz. That's where I varied it. That was the variable that was changed. The chemical characterization, where we used pH measurements, because many other papers use pH measurements to characterize it as the first standard, and then other different ways to do it. And then analysis through emission spectroscopy using a spectrometer from 300 to 800 nanometers. And that was the range for a lot of N and O excited species. That's why that was the one of focus. But you'll see as we continue on, there can be research in terms of the UV range, which what we want to look at maybe further in terms of further research. So finally was the biological characterization. The plants were grown in a controlled setting, so we can try. So I think if you've seen my presentations before, I've done research on agriculture with plasmas. So this is the most controlled setting we've had. So growing it within a grow room and using a hydroponic setup to reduce variability. So you can control the system, you can control the light, you control the temperature, you control the nutrient solution. And then we did an analysis of plant growth every 2 weeks. So when we did the treatment, I'll tell you what the treatment is. Every 2 weeks, we took measurements and we took photos to do comparisons after the entire experiment. Finally, the oil was extracted from these plants and then analyzed using DPP antioxidant assay in a spectrophotometer. And then we analyzed it using 2D GCMS by a different lab in Dr. Snow's lab. He was able to give me some results, and then further research is going to be done with the oil as well. So what was the treatment protocol? What do we focus on? So this protocol consisted of a 30-second treatment using helium gas plasma jet. So the helium gas was the basis of the plasma jet on the roots and stems separately. A standoff distance of about 3 to 4 centimeters was used, and the jet was adjusted accordingly to the size of the plant. So I'll explain how all of that's done in terms of the characterization when we get to the results, and you see here the photos of the different types of treatments. So results and discussion. Let's get to the results. This is most of the important information in terms of it. So the physical characterization, not much to garner in terms of the information from it. The plasma jet was constructed using a 1-inch long steel tube nested inside a 14-centimeter long borosilicate glass rod. That borosilicate rod was used as a dielectric to create that DBD jet. So you excite the actual long steel tube, and then the jet will be made inside that tube, inside the borosilicate tube inside of that. And then the cover is made of a 3D printed PLA as shielding to insulate the outside of the jet so we can move it up and down without just shocking ourselves immediately. The jet was mounted in an upright position for treatment. So this is how I describe the treatments here. That's what you saw from the photos. On a variable length stand, a variable AC plasma driver is connected to a steel tube, the steel tube that I just discussed, using a high voltage wire. And helium gas will be flown above. So you see here, be flown above here, and connected to the borosilicate tube via this inlet. So what were the characteristics we saw in use? Again, you see more in the electrical, not in the physical, but there are some characteristics in physical I just wasn't supposed to analyze in terms of this research. So the jet produces a plume effluent that is purple, relating to the oxygen and nitrogen content present in the atmosphere, mixing with the helium feed gas. Jet length and plasma regimes are variable from frequency, flow rate, and voltage, right? So they're just saying that if you change any one of those, you might see a change in how the jet looks, maybe how long it is, maybe if it's stable in terms of that, and then that's a whole different research in itself. So as I said, I showed you previously diagrams of how DBDs are set up. This is how our DBD is set up. The DBD is set up with a floating ground, and it's grounding out to the air. So before you saw that it would have a high voltage electrode and a ground electrode. Instead, it's grounding out to the air, which makes it unique to other plasma jets that you'll see in different papers in terms of plasma jet generation. You have the helium gas input, the same Teflon tube we just talked about, and then it's connected to the oscilloscope for electrical analysis. What did we see in the electrical analysis? So this is just a summary of what we saw with some of the values here. We're going to have more graphs to talk about electrical measurements. When the jet is varied in kilohertz, the voltage remains, the voltage RMS remains primarily the same as expected. However, the RMS current at peak to peak increased with increasing frequency. And then during plasma drops, this is plasma treatments. This is consistent for other treatments we've seen. The current and voltage drop slightly as the plasma grounds out to whatever it's touching. I think it's based on conductivity, but don't quote me on that. I'm going to do more research in terms of that. Here's some of the parameters in terms of it. You can just see it. I just stated most of it. We did 5 liters per minute for the helium, so it was mixing with the air. So moving further, right, I'm just focusing on the things we saw. So that drop we just discussed, the electrical drop. We did an analysis in terms of the oscilloscope to see what was actually happening. So here's the jet before treatment. This would be no treatment on the plants. Jet during stem treatment. So this is the stem treatment, and then jet during root treatment. And this illustration shows how the treatments look compared to the electrical results I'm going to show you. So this graph is supposed to illustrate what I just showed you previously in terms of no treatment, stem treatment, root treatment. And so the no treatment is a little higher than the root treatment and the stem treatment, which illustrates that there's a slight drop, not much there, as I discussed. So you don't have to focus much on that. But in terms of the current oscilloscope graph actually dropped a little bit more in terms of the root and stem treatment versus the no treatment. And you can see the root treatment is slightly lower in terms of it as well. So these oscilloscope graphs illustrated that percent drop in voltage, even though it was a little very small, and then the current peak to peak and RMS of the plasma before and during treatment. So that's how you characterize it, those electrical measurements. And so if that doesn't make as much sense in terms of the graph, I have the actual measurements here, and I'm illustrating specifically the root treatment because the roots are a focus in the study as well. But there was a drop in the stem treatment. And the average percent drop, so the jet's electrical characteristics during the root treatment featured an average 4% drop in voltage, while the stem treatment featured an average 2% drop. That's why I said the focus are the roots, because there's more of an effect going on in terms of the electrical drop. So to begin with that, now we focus on how I varied it from 26 to 30 kilohertz. So that's just the stem treatment, or that was just the treatment protocols there. Now we're trying to look at the characterizing of the jet in terms of if there's any variability in terms of electrical measurements. So the voltage versus frequency of the APPJ, and we saw here that there's not much again in terms of the voltage changing. It's not changing that much. But when you get to current, it changes about 4%, so 3.58% peak to peak. Overall, that was the average percentage change. And then for the RMS, it was about 6% in terms of the average percentage change as well for the current. And that was all the electrical measurements that characterize the jet. I don't discuss much of it here. When we get to the discussion section, we'll focus more on identifying what these effects might mean, or what we saw, what the result might mean. So now we move into the chemical characterization. Here, I put the whole spectrum in. Maybe I could have split it up to see more, but I identified 19 compounds through emission spectroscopy from 3 different spectra that I overlaid on top of each other to see if there was any differences in terms of them. There weren't, and there was a threshold to actually choose which ones were chemicals and which ones were just a part of the, as you can see, the small little divots in terms of it. Those weren't actually chemicals. And so of these 19 chemicals, we identified a lot of them. I think there's about 6 I didn't identify. And the spectra confirmed that excited species of nitrogen, oxygen, and OH are present in these plasma. These can have a very short lifetime. As I said, reactive oxygen species have a lot short lifetimes and can only be reliably detected through spectroscopy. It could be other types of spectroscopy, but emission spectroscopy for atmospheric plasma is one of the gold standards in terms of identification. And so from these different chemicals, I don't talk about it right now because I'm going to identify them on the next page. There are these different excited species and what they might be, or the different spectral lines and what the excited species may be. These are the actual excited species from those spectral lines. So these chemicals are a few of the species that were able to be detected through emission spectroscopy. However, there's others that are identified in literature that are probably present but are much harder to detect, or created due to reactionary processes from other excited species, and therefore aren't detected in the time we're actually running our system for detection, right? And so what are these species? As stated, the excited species themselves can eject electrons. If you remember when I discussed it before, that's what excited species is from pending ionization as well, and cause other excited species to be made, which would initially all be produced because of this creation of the jet. So the spark of that creation of all of these reactive oxygen and nitrogen species is the start of the creation of the jet. On the left are some of the chemicals that would be produced due to reactions from the species found through spectroscopy. And so here we got hydrogen peroxide, H2O2, so it's the reaction between the hydroxyl radical and hydroxyl radical. Then we have the superoxide, which is also the reaction between singlet oxygen in terms of its reaction. And we have nitric acid. So I'm not listing the ones we talked about already. Nitric oxide, which is just one of the excited states of nitrogen. Peroxynitrite, which is nitric oxide combining with the, or is it superoxide? And then nitrites, which can be created through the reaction of NO and O3. And O3 is present, it's just not, we can't detect it in terms of the spectral lines because they don't have one in the region we're detecting. So the focus was to try to identify some of the excited species. And now we're going to see what that species translates to. What effect does it translate to? And so for a lot of plasma researchers, in terms of these ecological effects, they try to characterize the characteristics of the RONS species by doing a pH measurement, then followed by an ORP measurement, and then if you can, identify each species by itself. So I was able to do it through pH measurements. And this graph illustrates the change in pH versus the activation time of the jet using a pH meter in 40 mLs of water at 3 to 4 centimeters away. That was the jet distance, similar to what I use for my treatment. And so you see here, it begins to go up and then go down. And then that's characteristic you'll see in other research that actually happens. And it might be due to the contaminants in the water. Not sure yet. More research needs to be done. So it's about a 5.83% change at 6.78 pH. Okay, so I'm just giving you the results. And now here I'm starting to integrate different papers and what you might see in other papers, right? This is a very good paper here. And I created a graph with all the data of pH versus activation time, similar to what I have here. And this graph just illustrates a comparison of the pH change and activation time of our jet to various other atmospheric plasma jets in papers and their respective pH and activation time. And so it's from Thermidus. He does a lot of research in just reviewing papers and trying to compile the papers together to try to get some sort of understanding, which at the end of the day is what science is all about. And so our comparison to these different pHs shows that it's a very small pH change. But as you see in other papers as well, this small change can be contributed to other effects on plasma agriculture as well. So that was our chemical characterization through pH and spectroscopy. Now we move on to the biological measurements. So here plants were selected from the middle row of the hydroponic setup for week 3, 5, and 7. So the first week I just took a picture of the photos. I shaped it down because they all look the same at the beginning. This green indicates that these were the plasma treated, and then I took them out. So A and C are treated, where B and D are controlled. And my just comment on it is that from week 1 to week 5 are probably where optimal plasma treatments are going to be, because at week 7 during harvest stages, you're probably stressing the plant when it's supposed to be trying to like end its life cycle, which isn't that good in terms of plant health and antioxidant activity. So moving forward, I just discussed the plants so you're able to see them. They're just visibly a little bigger in terms of that. But the focus right now was the roots of the plants, right? So the photos of roots of groups A and group B. Group A was treated and group B were control. And the roots are much thicker on the treated plants and generally look as though there's more radial and length growth in terms of it. And now that's going to be discussed more with other papers discussing what might be going on and why are the roots different in terms of treated and control. So these are the graphs where I compiled the averages together using Excel after taking all the data. And this shows the stem length of the treated and controlled plants. The graph illustrates that most of the percentage change was at the end. So the average percent change of stem length of treated versus controlled plants per 2 weeks was 8.97%. But on the root graph, you see that there's a gradual increase over time. And I think that's why I'm more or less focused on it as well. And it's about 10.66% per 2 weeks versus of the treated versus the control. So that was most of the biological measurements of the plants during growth. Now here is the biological measurements of the plants and its extracted oil. The amount of oil produced wasn't so much a focus, more or less the extracted oil, antioxidant activity. That was more of the main focus in terms of that. And then I showed here that I did 2 different extracting methods. They're the same extraction methods, but they had different recovery methods, which provided more oil at the end of the recovery method. But they were both done for each group. So that's why I lumped them together, and you can see the differences in terms of it. So this is not really that much of focus here in the average because there's a large discrepancy, but it's there for you to see. Okay, and it just illustrates the color of the oil in terms of it. So these are both treated. They're just a little bit darker. I'm not sure in terms of it, but that's not the focus of the research just yet. But antioxidant activity does relate back to the color. That was a big thing. The difference between the plasma treated. So here we did an antioxidant testing of the oil graph. Of the oil. There was no graph here, so I deleted the graph beforehand because it didn't really convey the message I wanted to say. But the antioxidant activity was done using the UV-Vis spectrophotometer at a certain thing, and DPPH is a stable radical, so it reacts with the oil, and you can do an actual good antioxidant test of it. There's other ways to do it, such as T-bars and other methods, but this was the focus because it can reliably convey the different antioxidant concentrations, different antioxidant activity at different concentrations. And so the difference between the plasma treated and control plants average antioxidant activity was about 2 to 3%, and you can see it here clearly at different concentrations. They have different antioxidant activity, and it's all based on the concentration of the molecule inside there, so there's some variability, but not much. And the first plasma-treated plants had the highest activated antioxidant activity average, followed by the second plasma treatment, and then the commercial sweet basil extraction, right? So I did an extraction with sweet basil, then these are the plasma treatments. I did a comparison to BHT that was already there as a standard. That was the antioxidants. Now finally we get into a spectra of GCMS, full spectra of, or full data set in terms of it. And this illustration presents a full 2D GCMS of treated and control groups of extracted oil previously mentioned. So compounds were able to be identified from this point that match specific organic compounds found in basil. I don't talk about this much because this research is still ongoing. I don't want to say too much about concentrations in terms of identification of peaks, but we were able to identify the peaks. We are looking into concentration. And here are some of the MS graphs that we got from it. So it's the graphs, yeah. MS graphs that we were able to get from it. In all samples, these organic compounds were identified in different concentrations with library hits of over 900. Eugenol, esterol, eucalyptol, alpha-pinene, alpha-ocimene, terpenol, etc. Estimated concentration of each component is ongoing, so we're going to see what we get from that from further research. These are some examples. So I showed eugenol here with the treated peak, treated sample that was analyzed in an MSGC with a library hit of about 940, and anything over 900, I was told is very good from the chemist who ran it on terms of it. And then that shows you that they identified because they reject the peaks under 900 in the system. And so what did we see in terms of everything? What were the results? How does this help us understand what effect is going on? So this discussion aims to shed some light in it. So even if you didn't understand most of what was going on beforehand, this will give you some background in terms of what other papers say, or what other people say, and how it can be related to the characterization of this jet. And so I said we saw a 2 and 4% drop during the root and stem treatments respectively. But how does that translate to the chemical things or chemical effects we saw, chemical species we saw? And how does that relate to the pH change as well? So a pH of 6.78 after 20 minutes of activation time translated to a pH change of about a percent change of about 5.83%. And the RNS species, as we discussed, that were produced were OH minus, O minus, N plus, N2 plus, and the helium metastable, and they were all in excited states, which leads to the chemical formation of other species such as I discussed, NO, NO2, and O2, which are the superoxide, nitrate, and NO. These reactive species most likely lead to this pH change because in literature, that's exactly what it's attributed to. So that's well known already and studied. Okay, and so how does this plasma treatment now relate to the biological measurement? So we just characterized a little bit of that effect in terms of how the pH gets to where it is from the plasma treatment. So what about the biological measurement? So the plasma treatment was characterized as creating RONS with concentrations that were able to create a pH change, as I said, about 6.78. So using the same treatment conditions with a different activation time, about 30 seconds, the roots and stems were treated separately. The treated growth rate was about the treated root growth rate was about 10% higher than the control roots, while the stem treated growth rate was about 8% higher than the control stems. It seemed like the presence of water, and that was just from initial investigation as well as prior research, people discussing how water is getting uptake by the roots a lot more. There's something with the water, and so it seems like the presence of the water on and inside the roots during treatment provided change to them that led to a higher voltage drop when the plasma ground into it. Does that relate back to the actual growth rate increase? Don't know, but from other research, we're going to garner some type of image from that. So it's noted in the literature that the presence of water provides a different environment for chemical reactions to take place. Specifically reactive species, as well as changing the effects seen on biological organisms, not necessarily increasing the growth, but increasing the small components we just talked about, such as eugenol and esterol. That was the big focus in terms of the research. This further led to thinking that different chemicals, which can be considered environmental oxidative stresses on the plants, can lead to different effects. and also localized area of the plants as well. And so I guess I didn't talk about this picture. This just shows the chemistry involved in water versus in air. So you might have some reactive species in air, and as it gets to the water, you'll have the creation of maybe aqueous HNO3, these different nitrates, and so forth. And as we get to this further thinking, that different chemicals, which can be considered environmental oxidative stressors on the plants, can lead to different effects on growth in localized areas of the plants. So genotype, this is a biological term, is just saying that you have a certain pre-programmed disposition to do something. But in plants, they have a lot of different genotypes that can be expressed as phenotypes. And based on environmental conditions, specifically oxidative stressors, they can change what's being expressed in the plant. So you can have a different amount of reproductive output, you can have a different amount of vegetative growth, and as you'll see, you can have different changes in antioxidant effects, which are common to the plant defense in terms of oxidative stressors. And so for the vegetative growth, that's also another fact, reproductive growth. We didn't really look at reproducing the plants, or maybe the flowers, but other people have in terms of flowers and fruits. And so continuing on, we just discussed what the thinking was and why we were going down this path of discussion, is that we saw an average 8% change per 2 weeks and then a 10% change on the roots. So that was 8% for the stems, 10% is on the roots, and the extracted oil was relatively low. So we didn't get much information from the amount of oil, but we got information from antioxidant activity, which showed that the first and second were the plasma treatments, followed by the control, and then followed by the, not followed by the control, but followed by the commercial plasma extraction, right? And so the second and first control were fourth and fifth, and it produced about a 1 to 2% difference in antioxidant activity. Just keep that in mind. I don't know if I had that here. I should have put that as an illustration, but that's what it was, a 1 to 2% change here. You can see from the numbers. And then what did the GCMS show? So the GCMS gave us the ability to identify 6 or 7 of the 15 compositional components in basil. These compounds of importance were eugenol and estragol, as they constituted the main composition of the basil plants, or the main composition of the 15 components we just saw. Concentration analysis is currently in progress. When we get to that, it'll be inside the master's thesis and hopefully be published in terms of that. And so now we continue. This is more of a discussion from things I saw in literature and how it might relate to our research. A small pH change, and how are they related? So how are they related to each other? How is pH related to everything? pH change was about 5%, correlated to an 8 to 10% increase in plant growth rate. That's what we saw. Okay, it isn't of cause and effect, we don't know. Of focus was the roots of the basil plants, as they looked much healthier and had a 10% growth rate increase. So how do pH and antioxidant activity relate? So plant health and growth correspond to antioxidant activity within a plant. Antioxidant production, however, can be stimulated by environmental parameters. The RONS created, this 5% pH change, would also be correlated to the average antioxidant activity being the highest at 81.98%, where the control plants had about a 1 to 2% average decrease. And that's a lot in terms of antioxidant. And here, this illustration just shows some of the stressors, environmental stressors in plants. They produce a RONS/oxidative burst in the plant, and then this creates antioxidants, different gene expressions, and osmoprotectants, which overall create a stress tolerance within the plant. Okay, that's what the further thinking leads to. And now RNS and growth rate. So we saw pH and antioxidant, now RNS and growth rate. RNS species are short-lived, but can have effects such as cell division and cell signaling, as we discussed, which can correspond to growth rate. The growth rate of the roots remained higher throughout the experiment and showed a healthier nature in photos. They had a higher growth rate than the stems. These are the two evidences we just saw. It's hypothesized in papers that the addition of liquid water to the gas phase plasma can change the resulting chemistry of RONS species being created. That's another big thing we just discussed as well with water and plants. And so again, RNS and growth rate, the plasma jet produces small amounts of RNS, which are known environmental factors that can cause a change, such as increase in antioxidant component of the plants we just discussed, specifically basil. As the plants were all grown together in the same conditions and the only external factor was the plasma jet, this leads to the relationship of the plasma treatment creating RNS which produce the desirable effects on the plant relating to growth rate. And this just shows biotic just means plant stressors that are alive. Abiotic just means environmental stressors. Plants get stressed out, they produce their own ROS. This produces oxidative stress, which can create these low weight molecular antioxidants, which are what and high weight molecular antioxidants, which we're measuring for antioxidant potential. But just remember, abiotic stresses can be RONS species themselves, and that inhibits that response there as well. And so now here we compare to real papers in literature. I was just telling you about the papers I read, but these are specific effects I'm noting to our characterization. First paper is the effects of plasma on plant growth, development, and sustainability. Second one is emerging plasma technology that alleviates crop stress during early growth stages of plants. And finally, we see reactive oxygen nitrogen species produced in water by non-equilibrium plasma, increased plant growth rate, and nutritional yield. So these are very specific to what I am doing, which is why they're the focus. So the first one we just discussed, effects of plasma on plant growth rate, development, and sustainability. So this paper investigated the effect of plasma treatments on the roots of wheat. The results saw an accumulation of different RONS species, which disturbed the formation of specifically microtubules in root hairs by chemical signaling. Their conclusive notes focus on how using plasma treatments as artificial sources of minuscule RONS concentrations, you're able to regulate different processes. And they also stated this is not only limited to the roots, but other parts of the plant as well, such as flowers or fruits. And it's illustrated right here. This illustration from paper one discusses the RNS species being used on different parts, such as on the flower, and it shows how the RNS species signals in a plant and how it can be uptaked through a plant, through the root specifically, because you're uptaking water. If you use reactive water, things can happen. Or plasma activated water, things can happen. Next, paper two focused on how specific papers, so they reviewed the specific papers and they focused on how the specific papers they reviewed related to plasma treatments. It discussed how DBD induces plant acclimation to stresses by inducing gene expression. So remember when I talked about how plants have different genotypes in there and different expressions can cause different things to happen, and chemical signaling. So gene expression is just the same thing as chemical signaling, or how I see it is the same thing as chemical signaling. A specific paper on wheat plants in this one talks about the chemical signaling promoting an increase in activity of antioxidants and defense-related enzymes within the plant. There are a few papers that discuss this effect on wheat seeds and seedlings. So remember, I'm looking at more full-grown plants or in juvenile plants. So it was talking about wheat seeds and seedlings, which was a focus here. And this one was still the, the crop stress and plasma treatment in terms of what it does in different stages of the plant. And then at the end of this paper, they talked about how from these different papers, they discussed how there's an optimum amount of time and power for when you do your treatment. Anything more, anything less will cause a bad effect or no effect to happen. And there were minimum exposure times of 0.25 minutes, which is about, I think, 15 seconds, and a maximum of 30 minutes in their review of papers. The power varied pretty widely from 3 watts to 300 watts. And if you saw from my table, our plasma jet was about 6 watts. So, we're somewhere in between there. Paper 3 finally gave me some good information because I was like, oh, why is the pH going up and going down? I saw that when I started the measurements and I was like, okay, I can't do it for 30 seconds. I might have to do it longer and longer to see when the pH actually goes down. So this paper was from Dr. Friedman and just looked at RONS species being used on different plants and what effects happened. And this paper gave a review of how the researchers characterized a plasma jet's RONS species in many different ways, including their pH. But the pH graph showed similar effects that I saw, where it increased in pH, then decreased at a different time step or different time rate. Okay, so theirs is much faster because they're using a gliding arc, which would have more energy and obviously create a higher amount of reactive species when it's generating. And so, yeah, this gave me a good indication that maybe this effect is actually what it is in terms of RONS species and pH. So finally, what did we all see from this data? So the jet was characterized through electrical measurements, RONS species present, availability of pH change, and the comparisons with other different plasma jets and treatments in the literature. The effects seen on plants that we identified in prior research were able to be described, and this study helped to shed some light on the viability of repetitive plasma treatments. So remember I said our treatments was every 2 weeks when we did it. We saw that there were no other papers like that, so we wanted to see what would happen, and it showed that increase in root growth. So just remember that in relation to other studies, most people don't do it like that. The investigation of RONS species and their effect on biological organisms conclusively showed that RONS could play a major role in all of different types of plasma agricultural treatments. So that umbrella term I discussed at the beginning is attributed to just how a plasma jet treatment does stuff, but now we can say maybe it's just mostly the RONS species doing what it does. And we know UV light does bacterial inactivation, but with the other effects, it's mostly the RNS species. And it can play a major role in the plasma agriculture. While this is already discussed in literature, this study was able to characterize some of the different effects seen specifically in RJET and attributed to the RNS species and other RNS species we discussed in literature. So through chemical extraction and essential oil analysis, the relationship between RNS and induced antioxidant activity was investigated, while most literature focused on these effects and how it relates to plants, like I discussed. They talked about how you actually can do that for antioxidant activity increase. This study was able to go one step further and float the idea of using RONS species to induce an increase in antioxidant activity in herbal plants for use in industry. This is beneficial, as mentioned, because of the many uses of antioxidants in the food and medicine industry. If there's any way to increase the effectiveness or the concentration of natural antioxidants, that'd be very useful in industry. So further research, as I discussed, is determining concentrations of those small components, those 15 components, including eugenol and linalool esterol. Then investigating if RONS species and specific ones, maybe it's superoxide, maybe it's nitric oxide, whatever it may be, and investigating them using ORP for plasma activated water, and then UV vis spectroscopy because NO lines are present in the UV range. And then finally, linking to see if there's specific components being changed from specific RONS species. So not doing such a general form I did where I can identify them, I can identify certain concentration of things, but being able to specifically look at if, let's say, superoxide ion can change the concentration of specifically eugenol in that sense. So I want to say a special thanks to the entire physics department for my journey through this process in terms of doing this study for the last 4 or 5 years. Dr. Snow did all of the MSGC analysis, him and his team, so I want to say special thanks to them. Dr. Tanya Lian was integral in making me understand how the chemistry was involved in terms of the antioxidant activity, because linalool isn't a phenol, so other different components in basil can be antioxidants. Dr. Lopez was guiding me in techniques of what to do, what to research, what to look up, do a lot of literature research, try to find as much literature as possible to make sure you know what's going on. Dr. Sahiner was giving me the opportunity to teach as a grad student and helping me along the way while I was doing this research. Father Jerry gave me a lot of the understanding and what to do for analysis of antioxidants, because I was shooting in the dark at the beginning, then it all came together. And then members of the stockroom and lab services, and specifically facility engineering, for helping me to make the grow tent and having the setup there to be able to do the project I wanted to do. And I think overall it's been a good learning process, and if people decide to pick it up in the LEAP lab, it's not as laborious as long as you decide what project you want to do. Don't go in there thinking I'll do whatever it is. Just think about what project you want to do. If you come with an idea, I'm sure they'll be able to help you. And thank you for coming to the presentation. Okay.
All right. Thank you, Will.
Yep.
Thank you. Thank you, Will.
So what we'll do is we have a few minutes.
If you have any questions,
we'll ask the committee, the thesis committee first. If Dr. Sahiner or Father Jerry have any questions, they can ask them now, and then we'll open it up to everybody.
Yeah.
I have a question, but I'll wait for Jerry.
Okay.
Oh, thank you, Alper. Yeah, Will, I just want to say, yeah, very good presentation. You collected a lot of data, and I think you really, the interesting thing, you really talked a lot about the RONS and their effect, how, certainly they're quite reactive, but also then, because of that, the activity that now is presented to the plant and into the oil, in terms of, let's look at the lipid oxidation reaction, something that's very explosive and radicals and all that. The question I have is these species, the oxygen, nitrogen species, reactive species, how would you see them kind of working together with the something like an oil-based or lipid-based
so you're saying
oil.
for the how would it work in terms of the oil-based, lipid-based foods, or just specifically?
Yeah, certainly in the essential oil of basil or anything, because you are treating the plasma is right.
Oh, so you're saying how are you go more in depth in how the antioxidant activity is being changed, maybe in terms of with the plasma treatment, right, with the oil and all that stuff? Is that what you're trying to say? I'm just making sure.
Yes, that's
okay.
Right. That's right.
Okay,
Yeah.
so I can explain a little further. I'll use an illustration, right? So, if you're a biologist, if you've taken any biology, if anybody's taken biology classes, right, there's a big thing in plants called hormones, but they're not the same as in humans. Well, they are the same. They're the same in terms of what they do, but they're not the same as what they are. So there's different hormones here. You see auxin, ABA, what's another one? JA. So ABA is abscisic acid. JA is jasmonic acid. SA, I don't know what that one is. I think it's like salicylic acid. But these in different concentrations promote that, like I said, the expression of a phenotype. And in these walls of these cells, in terms of cell differentiation, cell proliferation. So I didn't say much in terms of what was on this, but it is salicylic acid as well. But in cell proliferation, cell elongation, cell differentiation, these concentrations play a role, and then these RONS species can react with the concentrations changing what goes on. So it might be a positive at certain times, and it might be a negative. And then furthermore, when these concentrations change, you lead to this right here. It's this one, and then let me see if I have one more. Anyway, in this paper, they talk about how, is it this one? Oh, it's this one right here. Yeah, this right here. So in this paper, they talked about how there's two different defense mechanisms in plants for when RONS species are just being present. As I discussed, any environmental stressor will produce an effect on the plant that makes it produce ROS species in low concentrations in the plant as a form of gene regulation or defense regulation and so forth. But if you introduce it to the roots, as they're discussing in this paper, it can be taken into the plant and have an artificial increase on it as well. So yeah, because that's the most, I believe, I don't know if you can do it on other parts of the plants, but they did discuss how, using plasma activated water, it's being taken directly into the plant, you're artificially increasing the amount of antioxidant that would normally be present if you have used regular water in terms of that, and you're not actually doing a plasma treatment. So you're just treating the water, then you're using the water afterwards. So that's somewhat what it's attributed to. I don't know in the stems. I don't know if it's the same thing. I don't know if it can go through, like, let's say, there's holes on leaves that are called stomata, I believe, and this is just where gas exchange goes on for CO2 and oxygen. So it could happen there as well, but they don't know, but they know for sure that there's been plasma activated water treatments, and then they use it for plant growth and have seen effect. So you can relate it back to that.
Yeah, good, Will. Thank you for that. And as you responded to the question, I think you made very clear how science research is so interesting, because you're talking physics, you're talking chemistry, you're talking the biology, whatever else. And so this really is a wonderfully collaborative example of research where you're bringing in so many groups. I would share with you that this research in time hopefully could lead to additional natural antioxidants, because of the difficulties with the synthetics.
Yes, so that was the focus, right?
Yeah, your GCMS data hopefully will shed more light on that. I'm sorry to take one other question, but I think your point there in the acknowledgments for Dr. Lopez is that I just want to say, not that I have a keen eye though, but just your over the years now, how your appreciation of, let's say, science research or your growth in that has been really quite significant. So I would applaud you on that, and applaud you for a very good study in terms of bringing in more of the control of the use of the plasma.
So we
and
saw
all of that.
right. The previous research was less controlled to see if there was anything going on, and now we tried to get it more controlled so it can be publishable when it's done.
But that data, the the pictures of the roots, the
and all that
stems
stuff
are very, very impressive. So but thanks so much. you.
Thank you, Will. It was a great presentation and a great job. A couple of questions for you. First of all, I mean, I saw from the results from the control versus the plasma treated one, it's obvious that the stems grow very well and those things. And one thing that you said that in one of the slides, after 5 weeks or something, the plant goes to a kind of a reactive position or something, week 5 and 7. What's going on at that time? We have to stop at 3 weeks or something, or
so like so I gave a little discussion about it as well. So remember, I'm doing repeated treatments, not similar to other papers, and then so this optimum power and exposure time doesn't take into the account that most of these papers are doing research on plants that are in juvenile stages / seedlings. So when you step it up and you look at like crop stressors in plants, or like I had to do a lot of research in just plant biology and plant ecology. Plants themselves at the end of their lifetime are more prone to effects on stress, like bad effects in terms of stress. And so when you have the treatments, and this is how I explain how I think these photos might be going, or what's happening, is that when you have those
lifetime for a basil is about 7 weeks, 6 weeks, something.
It's about 8 to 9 weeks.
I see. Okay.
Right. And so I harvested at 8 weeks, and then I'm saying that these juvenile plants have more. I don't know if it's robustness, but they can recover more. And as you're stressing them out, I think from week 3 to 5, this is more or less the middle of the life cycle here. They still have a little more stress tolerance that's already present as well from the plasma treatment, but already in the plant inherently. So the seedlings that just have more stress tolerance than plants at the end of their life cycle. And so that's why when we did the stress treatments as the plasma treatments as a repeated cycle in terms of every 2 weeks, overall, at the end of that life cycle, they might be more prone to damage at these concentrations rather than positive effects being seen.
So there's an optimization factor of the exact time for that. Second question is about the, I mean, there's a lot of chemistry going on. Of course, you start with the plasma physics, but then what's happening is probably the most important is the pH levels drop, and also the oxidation effects. And in the first, when you're saying at the end of the slides that there's a rise in the pH levels for the 30 seconds and then goes down. What's happening that why does the pH level rise?
So I explained a little bit. I did a little bit of research in it. I don't think many people are doing research in that, interestingly enough, but it could be. So I discussed with Dan a little bit. There's a thing called solvated electrons. I've never researched that a little bit, but it's like one of the highest oxidative solutions, and that can be attributed to that, as well as different anions that are already present, or cations of different we call it metals or whatever contaminants in the water being changed from the reactive species being produced from the jet. And so when that happens, it just takes some time for that to actually change the water and then finally stop reacting with whatever's in the water and produce that pH change with the water to create that nitric acid.
Okay, and the third question is about, I mean, your plant is chosen for this one is the basil, I guess, with basil.
Yes, it is.
Do you think that it's going to be quite different for different fruits and vegetables because of the chemistry is quite different for them? All right. So why did you choose basil for that? Is this a more controllable sample or something?
It's because, so we did a bunch of plants back then, right? So what information were we able to get from it? We were able to get growth rate. Maybe if we go to the full term, we can harvest the plant and see what's coming off of it. But that's a limited amount of data, and we were able to look at different herbal plants because they're used as extracts afterwards, and you can measure actual antioxidant activity. Furthermore, I mean, like doing the research, there's a lot more prior research behind our basal. We did like someone else did research into it. Someone else did research into it. They found different effects. So we're compiling on the evidence that's already present, as well as that we can use the information here much more. There's more information you can garner from that and the extracted oil rather than if you were growing, which is an example. We did kidney beans, we did sorrel, we tried different other plants, but you're not getting as much information than you're getting here to relate RNS species back to antioxidants. So if we did that same treatment on something else, how would we measure antioxidants activity in terms of that?
Thank you. Thank you. Very good answers. All right. Thanks.
All right. So that's everything. If anyone has any more questions.
So, Will, thanks. I'm going to yield my questions to everyone else because we've spoken a lot over the last few months on this topic. So I echo Father Jerry's point. This project is so many different areas. It's on the periphery of physics, chemistry, biology, even engineering, building the grow tent and everything, and the hydroponic system and everything. So yeah, very impressive work, Will, doing this. So is there anybody from from our participants, attendees, that would like to ask Will any questions? Please just raise your hand, and I'll call you. Lisa.
Okay.
Lisa Rose-Wiles, Dr. Rose-Wiles.
I'm sorry, I was muted, the muting thing. So, Will, I'm so happy to see you continuing with this basal work because I remember this, what are we going back, 4 or 5 years? How many years are we going back now?
Three. Not that far back.
3? I thought it was longer.
You guys did a paper too, though. So your paper, Dr. Lopez.
It's maybe going on 4 years. You're right. It's going on 4 years. Yeah.
Okay.
But I'm remembering, I think it was the first year when you were working with, or Father Jerry and Jose were working with basil plants in the greenhouse. And some of us were lucky enough to be given some of those basil plants, if you remember. And this is just a little anecdote, and I don't know if it's even worth exploring. But as I'm sure you all know, basil, like when the first frost comes, it's gone, right?
Yes.
It's very vulnerable to frost. Right. So I took my basil plant inside before the first frost and I overwintered it, and it came back the next year. It didn't come back the following year, only 1 year. I don't know if this is anything to do with the treatment, but it's the first time I've ever managed to overwinter a basil plant. So I don't know if that's anything worth exploring, because we all know, basil is really fragile, and it's an herb that I absolutely love. So I just thought I'd throw that in there as a comment and a thank you. And
well, thank you.
So happy to see this work continuing.
I'm hoping someone else will pick it up too, in terms of it.
We have been asking questions about is anything genetically passed on to the next generation? So that was a big thing to know.
Yeah, is it changing the entirety of the plant? Are you increasing stress tolerance for next generations and so forth? So maybe that's a whole region of study as well.
Yeah, I only had the one plant, and it only came back 1 year. It didn't come back the following year, but that may have been my bad gardening practices.
Thank you,
Well,
Lisa.
thank you for the comment.
We have another question from David Turner.
All right, David.
Okay, I actually wanted to ask about the genetics aspect of it, because I remember there was a study. I probably should look more into it, but apparently there was like certain traits that you wouldn't expect could be passed down in generations of mice, such as like maybe the fear of like electric shocks or like certain colors or something. And I guess plants are kind of more simpler than like I guess animals. So I was gonna ask about whether or not the traits that were given to it by the plasma would be passed down to the next generation, but it kind of got answered before.
Oh, so it did, yeah. So I was like, we don't know for sure. That's a whole field of research there. But I mean, initial research stages say no, because RONS species are environmental stressors as themselves. They're produced in the atmosphere. That's a whole. Medical research looks at free radicals. Free radicals are what RONS species is. Antioxidants are also being studied as well. That's a whole thing. And so when it comes together like that, yeah, you could find papers relating back to it, but I'm not sure.
Okay.
Maybe if you look more research into it, you'll find something.
Because maybe so much not as like genetic, but maybe more chemical. Like
right, it's more of a
I know, like
change.
if someone's obese and they're pregnant, the child has more of a chance of becoming obese. So I thought maybe it was something like that, maybe perhaps.
I see.
So we'll take one more question from Professor Schwan, Martha Schwan.
Okay.
Yes, if I can unmute and on camera.
You're unmuted. You're unmuted. We hear you.
Thank you, Jose. For one thing, in warmer zones than ours, basil is a biennial plant. So that's normal for basil.
Yeah.
I don't know if you noticed if there was, this is more from a geology standpoint, if the extra roots meant more water absorption, and did you notice if there was more effective use of oxygen in the photosynthesis of these plants?
I got you. So we wanted to do that. So prior research has done, we tried to do water measurements. Those setups weren't really the best in terms of like trying to keep that all stable and doing the measurements. And so with this setup, since we kept it all together, I can't definitively say for sure if it's using more water, but prior research from, I guess, I don't know if you knew him, he was the one doing the research, Sylvelson. He discussed how there might be more water being uptaking from the roots after these treatments. I'm not sure. He didn't do conclusive measurements. We did some small measurements that showed something with the system upstairs in the greenhouse, but I couldn't do it with this setup right now. But I'm sure there is something there. I don't know what it might be though. And then with the oxygen measurements, that'd be something to be able to measure that. But I don't think we have the capabilities to look at like photosynthesis for like the different reactions going on inside the plant cell. But that'd be something like if you were able to do maybe chemical tagging. You can tag different components of different chemicals and use them and see where they're being uptaked. If different concentrations of reactive species are somewhere, if you're able to get a chemical marker like that for reactive oxygen species. I don't even know if that exists yet, but yeah.
Well, these would be important for future agriculture, more photosynthesis, and also for infiltration and improving flooding, non-flooding conditions.
Yeah, that's very true. So you could use those roots to control the area in terms of the ground, keeping everything together. That's a very good question.
Yep. Thank you, Martha. Yeah, we've been discussing we've been learning a tremendous amount of plant biology, and we've actually begun a collaboration with the plant biologists at Rutgers University, who are the state plant biologists. But interestingly enough, what Will just talked about, we as physicists think of everything high tech. They're very, very low tech.
They're very low tech.
We learned a lot of stuff from them, but they literally look at plants
and still measure it with their hands.
So
it's all just the same. There's no robot.
Maybe we have to work with microbiologists or something like that.
For the paper we worked on, they were like using their hands to tie things together with rope, and just that was basically the study and stuff like that.
Yeah. So let's clap. Let's give him a virtual applause, or everyone. Will,
thank you,
congratulations on your work.
Oh, yeah, no problem. Thank you. Thank you so much for everybody coming.
So Will is not out of the woods yet. He still has to finish officially writing his thesis. But he's done a great job here. So thank you very much, Will. And
thank you,
we'll continue with our program.
All right.
So thank you, Will.
Yep.
So we want to continue with our program this evening. We are of course participating as we have for the last decade in the Petersheim Academic Exposition. This year, thank you, Father Jerry. This year we are celebrating at the university the 25th anniversary of the Petersheim. We thank a few people that are part of the committee, specifically Dr. Suli Chang from the Department of Biological Sciences, who's joined us tonight. She is the co-chair of the Petersheim Academic Committee. She's joining us tonight, and also Professor Weining Wang, who is a very active member of the committee. She's the secretary of the organizing committee, and also, of course, Professor Schoen, who I believe is one of the longest after Dr. Chang serving members of the organizing committee of the Petersheim, and of course Dr. Rose Wiles, who you met earlier today. Hi, Suli. I don't know if you'd like to say a word or two about the Petersheim or anything. Not to put you on the spot, but.
I say congratulations for the wonderful work. I actually enjoy very much the method defense, and I believe I attend one last year too. Very impressive, and very amazed that the oxygen reactive species, large species actually can have this broad application. I actually want to ask you that I didn't get a chance that can that one be applied for some kind of pattern?
Yeah, we are having talks about what we can do with that, and we have been talking about intellectual property and patenting, but that's a whole other story. At this moment, we're trying to figure out the science, as Will has done a great job of this time. But
Yeah.
yes.
Okay, thank you.
Thank you, Dr. Chang. So without further ado, I'd like to continue our program. And of course, at the heart of the Department of Physics are our undergraduate students, our physics majors. And for many, many years, that's been one of the strengths of our department, the tremendous amount of work that our faculty do in physics do with our undergraduate students. So we have a great group of folks tonight. That you all listen to. So our first speaker will give a 15-minute presentation, and Jasmine Emerson, and it's entitled Investigation of Plasmonic Resonance in Metal Nanoparticle Embedded Cadmium Telluride Cadmium Telluride Solar Cells. Okay, Jasmine, please. You should have sharing ability.
Yeah, thank you so much. Just give me 1 second so I can pull that up. Can you guys see the PowerPoint?
Yes, perfect. Thank you, Jasmine.
Thank you. So as Dr. Lopez said, my name is Jasmine Emerson, and today I'm going to go over the research I've been conducting for the past 2 years in Dr. Sehuner's Advanced Materials Synthesis and Characterization Lab. And again, like Dr. Lopez said, my research is entitled Investigation of Plasmonic Resonances in Metal Nanoparticle Embedded CDS-CDTE Solar Cells. Here is my outline of today's presentation. In my introduction, I'm going to go over the significance of this research, previous experimental research we've conducted, as well as the method of post-laser deposition. I will then go over my most recent research involving CDS-SI structural predictions, what we hope to accomplish in future studies, as well as my acknowledgements. Here are just a few reasons why our research is significant. First, we are creating alternate solutions to solving the world clean energy problem. Solar cells, as I'm sure you know, create renewable energy because they harness energy from the sun and they transform it into usable electricity. This electricity is produced with almost no pollution, making it very environmentally friendly. Second, we are working on the development of cheaper and more efficient solar cells. The current conventional solar cells that we tend to use are more expensive than thin film solar cells. However, they are also more efficient. Through our research, we are working on making thin film solar cells just as efficient as conventional solar cells, as the production costs are significantly lower. And then finally, we're building off of research that has been previously conducted by other Seton Hall students. In the past, these students worked on the effects of both silver and gold nanoparticles have on the efficiency of CDS and CDTE thin film solar cells, and they received very promising results. Through our current research, we are working on semiconductor comparisons to these previous studies. Samples embedded with gold, silver, silicon, and silicon germanium have been previously developed through the method of post-laser deposition. A Kelvin source meter was used to determine the electrical characteristics of each sample, made to determine if the added nanoparticles would influence the efficiency of the thin film solar cells. On the bottom right, you will see the results of the gold deposited samples. These results showed that the gold deposited for 60 seconds has the highest efficiency curve. In the middle, you see the results from the silver deposited samples. As opposed to the gold samples, these samples results shows that the highest efficiency curve is when the silver nanoparticles were deposited for 150 seconds. So while the gold showed that less nanoparticles had a higher efficiency, more silver nanoparticles had a higher efficiency. As a part of our current research, we focused on comparing the effects of silicon and silicon germanium nanoparticles embedded in these solar cells to those of the gold and silver embedded cells. Although these nanoparticles embedded into these cells are not metals, we believe that it will give us interesting effects, as well as give us a metal versus semiconductor comparison. Our current research originally only included silicon nanoparticles. However, we were unable to observe these deposited silicon nanoparticles on the experimental samples used when using scanning electron microscope, even though we were able to confirm that the silicon particles were present through energy dispersive X-ray spectroscopy. We originally believed that this was due to the deposited silicon being unstable, so we moved on to depositing silicon germanium nanoparticles instead. We did this in the hopes of stabilizing the silicon nanoparticles, and because adding germanium should in theory increase the charge carrying capabilities of the nanoparticles themselves. So finally, on the bottom right, we do see the silicon deposited sample. As shown in the image, the initial results showed a ohmic behavior. Although the silicon germanium nanoparticles within these dimensions are subject to photoluminescent behavior, this was not observed when the nanoparticles were deposited onto the thin film solar cells. While we don't have a definitive answer right now as to why this behavior was not shown, we believe that it could be due to the average size of the particles being too large. Through our computational and theoretical studies, we hope to determine why we were unable to view the original silicon nanoparticles in our experimental studies. So as stated earlier, the samples were developed in our experimental studies were generated with the method of pulse laser deposition. This is also known as PLD. This method was used because it gives us flexibility to create different structures due to all the parameters that can be varied, such as temperature, pressure, laser frequency, laser energy, as well as the time of deposition. In this method, a high-powered pulse laser is directed onto a target within a vacuum chamber. The laser beam first is focused through multiple cylindrical lenses before going into this chamber. And when the target is hit by the laser, the material is consisted of is deposited onto the glass substrate. A plasma plume is then created and condenses onto the substrate into a film. Using this method, we developed 2 half-size silicon germanium cells, 1 full-size silicon germanium cell, and 1 half-size silicon cell successfully. The half-size cells serve the purpose of being used for observation in the scanning electron microscope, while the full-size cell was used for the electrical characteristic testing with the Kelvin source meter. Shown here is a simple schematic of a thin film solar cell. Using the PLD method, we first deposited the CDS onto the ITO glass for 15 minutes. Afterwards, we deposited the desired nanoparticles for a very short period of time. For example, when we were developing the full-size silicon germanium sample, we deposited the silicon germanium for 60 seconds at room temperature. We deposited the silicon germanium for such a short period of time due to our desire to only deposit nanoparticles of the material rather than creating an entire film. Finally, we deposited a layer of CDTE for 156 minutes, making it the largest layer in our solar cell. So in this past year, we have done theoretical structural predictions, and they were made using the materials project. Using the information that was found on the materials project, we hope to eliminate unneeded and unstable experimental research by focusing on the most promising computational data that was found on the website. During this theoretical and computational research, we focused on the predictions of CDS and silicon due to silicon being deposited directly onto the CDS layer of our samples during experimental processes. So a phase diagram was constructed for cadmium, sulfur, and silicon. The diagram shows 3 predicted stable compounds, CDS, CDS2, and SIS2 as shown on the left side. From this information, we determined that CDS and SIS2 are most likely to be helpful in our research as they are neutral molecules, as opposed to CDS2, which is anionic and can't be used to easily form thin films. Observing this data from the phase diagram, we were able to determine the compounds that are most likely to be made when developing our silicon nanoparticle embedded thin film solar cells. The first compound and the one that we aim to create during our depositions is CDS, As shown in the schematic earlier, this is the compound that makes up the first layer of our thin film solar cell that we are developing. Therefore, we fully expect this compound to be made effectively, as it has in past experimental work that we completed. Using the most useful information that we gained from this phase diagram is that the compound SiS2 is likely to be made during our deposition of silicon nanoparticles. As I stated earlier, in previous experimental trials, we have been unable to identify individual silicon nanoparticles with SEM imaging. And with this information, we now know that this might be due to SiS2 being created during this deposition onto the CDS thin film layer. On the right-hand side, you'll see the Pore Bay diagram that was generated with the materials project for CDS and silicon. A Pore Bay diagram, also known as a potential pH diagram, depicts aqueous phase electrochemical equilibria. This really just means that it shows water-stable phases of the given compound as a function of pH and potential. Using this Pore Bay diagram, we have determined that the following compounds that are highlighted in red are most likely to be developed during our experimental depositions. And using this information that we found with the Pore Bay diagram, we were able to determine more acceptable parameters such as temperature and frequency that we should use during our PLD method of depositing the silicon nanoparticles. Shown here are the crystal structure predictions that were generated for both CDS and SiS2. CDS does have two different crystal structures. The one here shown on the left is the more stable structure and is the one that we aim to create during our depositions. This hexagonal word site structure is pyroelectric, meaning that its crystals are naturally electrically polarized, resulting in them having large electric fields. The crystal structure of SiS2, shown on the right, is one-dimensional and consists of chains of edge-sharing SiS4 tetrahedra. Again, we believe that we have been unable to view the individual silicon nanoparticles in our experimental studies due to this compound likely to be made. Overall, the most beneficial thing that we've learned in this past year from all this theoretical research is that this compound of SiS2 is likely to be developed when we are depositing these silicon nanoparticles onto the CDS layer. In future experimental studies, we will be modifying this pulse laser deposition parameters to synthesize silicon-only nanoparticles on the CDS surface, hopefully to get more promising results with and get non-ohmic behavior.
Thank you, Jasmine. So wonderful how you were able to do this work, even moving over to computational work and doing that. So we'll open up for questions from anybody, any of the attendees, any of the participants. Any questions? Okay. So you did such a great job, Jasmine, that everybody's stunned. So there's no questions. So let's thank Jasmine with virtual.
Thank you, Jasmine.
Applause.
Great presentation. Thank you.
Yep. Great job.
Thank you.
So our next speakers are Alexis Duffy and Ramiz Al-Jilat, and they are working with Professor Mitra Fazabadi. And they will be presenting a short talk, 8 minutes, with 2 minutes for questions. It's entitled Evaluating the Electric Charge of Kenzin-1 in Vitro. So Ramiz and Alexis, I don't know which one of you has a presentation. Do you have it, Alexis or Ramiz?
Yes, just 1 second.
With the tag team, I always wonder.
Hi, Ramiz and Alexis.
How are you, Professor?
Yep.
All right, can you guys see everything?
Yes, we can see.
Right, perfect. So good evening. I am Alexis, and Ramiz and I have been lucky enough to work under Dr. Fezabadi in her biophysics laboratory. And we are actually fortunate enough to be able to have this research ended up being published. So there is many more details in the published paper. So we're just going to do a brief overview, and we can also share a link to the published paper in the comments if anyone wants to take a look at it. As Dr. Lopez says, our title is Evaluating the Electric Charge of Kinesin 1 in vitro. Before we get into some more specifics, we need to first talk about molecular motors, which are proteins that are responsible for intracellular transportation and cell division, and they usually walk along microtubules. They interact with microtubules, and they usually have a positive charge, the microtubules those are, and they are bound together through electrostatic connections. In the cell, there are a lot of proteins, and in order to confirm the charge of molecular motors outside the cell, you need to do that through a nanotechnology-based approach. Thorough examination of the charge of the motors leads to better understanding of the nature of electrostatic interactions between motors and microtubules. The offered nanotechnology-based approach can validate the charge of motors in the absence of any cellular components. Our nanotechnology-based method will help us monitor their behavior inside a uniform electric field, and the approach will minimize the cellular functions that can interfere with evaluating the charge of motors. And just for reference, there are 12 different categories of molecular motors, but for this project, we specifically chose to focus on kinesin 1.
So the way that we started with this experiment is we had 1 microliter of microsphere beads that were 0.5 micrometers in diameter. And we had to dilute these beads about 4 times in a serial suspension of purified water to get the concentrations that we needed for the experiment. And in the experiment, we used 2 different concentrations of kinesin and beads, which were 0.25 milligrams per milliliter and 0.5 milligrams per milliliter. And we divided up these solutions into 2 different halves. Half of them were either used immediately in our experiments, or they were incubated for 30 minutes at room temperature, or 22 degrees Celsius. And this was just to allow the kinesin to be able to bond to the beads better. As you can see in the picture, we just put them in a small laboratory tube, and that's just how we mix them together. And when we were ready to prepare the slides, we put 1 to 1.2 microliters of this solution onto a nanocapacitor-based slide. And this nanocapacitor-based slide has an indium tin oxide on it that has a gap in the center about 150 micrometers wide. And the reason why this gap was so small was because we wanted the electric field or the force to be extremely high to allow these beads to move. So once we created the sample, we attached a DC power supply, and we used 2 different voltages in our experiments. The first voltage was just 1 volt, and that gave us an electric field of 66 volts per centimeter. And the other voltage was 1.5 volts, which gave us an electric field of 100 volts per centimeter. The reason why we didn't go much higher than 1.5 volts was because we didn't want to burn out the capacitor that was based on this slide. So 1.5 was about as high as we can go. To actually see these beads in motion after the DC power supply was connected, we used a dark field microscope with an oil immersion lens that had a camera attachment to it. So we would record videos of these beads with all the different combinations of voltages and concentrations. And we analyze these images or these videos in a program called ImageJ. So as you can see in figure 2A, that's just a picture of a bead in between the gap of the indium tin oxide before any type of voltage was connected. And in figure 2B, it's the same bead, but moved a certain distance. And ImageJ allowed us to track this bead and its motion and allowed us to figure out the displacement for it. And this was just the basis of our analysis that Alexis will now discuss.
Before we get into more specifics about the microsphere beads that are coated with the kinesin, we need to do a set of control experiments because it was particularly important to first monitor the behavior of these plain microspheres inside the electric field. Upon the implementation of the DC voltage and establishing a uniform electric field between the electrodes, the suspended microspheres and the surrounding liquid media started to move towards the positive electrode of the capacitor. This was an indication that the beads were originally negatively charged and they started to move due to the electrophoretic force. The observed displacement of each individual bead primarily resulted from interactions of different forces. The electrophoretic force created by the electric field on the originally negatively charged beads, and the drag force on the beads due to passing through the surrounding solution. As illustrated in Figure 3, the beads started to move under the electrophoretic force and were consequently affected by the drag force of the media, which slowed down their drifted motion, causing them to become completely immobilized or show minor Brownian motion. So Figure 3, we can see that these are just the microsphere beads that don't have any kinesin proteins added onto them. The red is shown at the applied electric field of 66 volts per centimeter, and the black is of the higher applied field of 100 volts per centimeter. And what we saw was under this higher applied voltage, the microsphere beads traveled an overall larger displacement. After we were able to look at the control of the microsphere beads, we are now ready to add the kinesin proteins. After we added these motors and observed the new coated beads under two different concentrations of the proteins, as well as incubated versus non-incubated, and as we increased the concentration, we saw that the reduction in the displacement of the coated beads. This reduction is more likely linked to the change in several factors. The presence of kinesin 1 protein in the solution elevated the viscosity of the solution and consequently increased the strength of the drag force acting on these beads. In Figure 4a, we compared coated beads to uncoated beads. The coated beads are shown in red and the uncoated beads are shown in black. And we saw that the coated beads, which now have the kinesin 1 added onto them, traveled a lesser displacement. This is indicating that the kinesin 1 is positively charged, therefore decreasing the force of the electric field. The beads, which were incubated with kinesin 1 for 30 minutes, they were more inclined to be coated by the molecular motors in the solution. So Figure 4b is showing the two different concentrations of kinesin that we attached to these microsphere beads, and these were both incubated for 30 minutes. The black is shown at the higher concentration of kinesin of 0.5 milligrams per milliliter, and the red is at the lesser concentration of 0.25 milligrams per millimeter. At this higher concentration of kinesin, we saw that it was the microsphere beads traveled a lesser displacement, leading us again to show that these proteins have a positive charge because it is decreasing the displacement once again. A shorter drifted displacement of the kinesin incubated beads as shown, as compared with the unincubated beads, is evidence that the electrophoretic force acting on the beads produced by the electric field was weaker. This change on the force can now be linked to the decrease in the net distribution of the original negative surface charge on the beads. This reduction can be caused by the positive charge that motor proteins attached to the beads carry, and this is an indication that motors carry a positive electrostatic charge. Each time that we moved another step in the experiment, we saw the displacement of the beads continue to decrease, which led us to conclude in proving that our method can detect and evaluate the charge of these molecular motors. The observations confirm that this approach can be considered a method to detect and evaluate the charge of motors, and it also confirms that kinesin 1 proteins carry a positive electric charge.
We'd just like to acknowledge funding from Claire Booth Luce for Alexis and from the New Jersey Space Grant Consortium for myself. And again, we did publish a paper on this research, so if anybody's interested, we can put it in the chat so you can check it out.
Thank you, Ramiz and Alexis. Great job. Thank you.
Great job. Any questions? Looks like a question from Will. Will Davis.
Okay, so I know you two. I just had a question. I think this was a great research, but I just had a question. So you chose a time to about 1.8, 1.6 seconds. I don't know if you mentioned, but why did you stop there? Because I see the graph increasing. I wanted to know if there was like plateauing at any point for displacement.
Yeah, so due to the viscosity of the solution, the microsphere beads eventually became stuck in the solution, or they began to just show minor Brownian motion. So after about a total of like 2 to 3 seconds, depending on the video that we were analyzing at the time. That's when they became stuck.
Okay, so you did mention that. All right, thank you so much.
Any further questions? So great job, and you have a published paper, and I see many of our other graduates there. Jane Breslin and Ibu also are on that paper, and thank you for acknowledging your funding sources. I should also mention that Jasmine Emerson is also a Claire Booth Luth scholar, as is Alexis. So as we know, both brilliant women in science, and hopefully they continue further. And then, of course, Ramiz also received the New Jersey Space Grant Consortia that Dr. Sahiner is the principal investigator for that particular grant. So thank you both. Excellent job. Keep up the good work. So we move on to our next speaker, who is David Turner. David Turner has been working in the Laboratory of Electrophysics and Atmospheric Plasmas. And the project that he was given was to start building, as best as possible in the COVID times, a plasma glow demonstration device. So the title of his talk is entitled Creating a DC Glow Plasma Discharge System. So David? David, you're muted.
Oh, sorry about that. I'm just getting everything together. Can you see me yet, or?
We don't see your picture. Now we see you.
Oh, okay. All right. Now let me share my screen. And then how do I wait, hold up.
Just go to presentation mode. At the bottom there, at the bottom left-hand corner, you see the one that has the excuse me, right-hand corner.
Oh.
Yeah, nope. Move one over. Keep moving. Move to the right. That one. Yeah, hit that. Like that. There you go. Okay, you're ready to go.
All right. So my project was to create a DC glow discharge system. Sorry, I'm a little nervous at the moment. So if I study that, that's what that is.
Sorry, take your time.
All right. So basically, the goal of this project was just to observe the properties of a low voltage plasma inside of a glass tube. We were going to use we're basically going to be discussing the properties of Pechet's curve. Primarily, I wanted to work with nitrogen, but I wasn't able to get that far through the assignment because we let me see. Hold up. Sorry. So yeah, basically, we were just gonna play with the atmosphere inside of a glass tube using a vacuum and a Brockhorst 7400 flow controller system, and but basically by manipulating just like certain aspects, I'll get to that later, but certain aspects of the environment of the tube, we were gonna basically just try and prove that Pechet's curve worked inside of the plasma generator. So basically what's Pechet's curve is basically in certain gases they all have a certain window of range between breakdown voltage compared to the pressure and space of the atoms. So like we have regular air, which is just I was working with in the lab, which is basically just 77% nitrogen, 22% oxygen, and then a couple of percent everything else. Am I right about that? Atmospheric pressure. So basically I'm sorry. So let me see. All right. So basically the breakdown voltage is it's when you're able to create like a what do you call it? Not electricity, but a plasma within a certain within the sorry, just give me a second. Okay, so basically we just be able to create a plasma between two electrodes at a certain point of in an environment of Well, can you just give me 1 second? Dr. Lopez?
Take your time, David. Take your time.
Take your time.
Okay.
So you try to create a plasma in a different gas environment, right? And you have the different gases there that you're showing us.
All right. So the reason it cuts off at certain points is because let's say you have let's say you're walking across a path, or you're walking through, let's say, like a room. If there's no floor there, if there's no floor there, you'll be able to fall through. And you're the plasma in the situation. But the reason why, if you have like a little bit of space but a really high pressure, the over here the gas will cut off, is because if you have, let's say, the floor starts to kind of, I guess, collapsing on itself, and there's just a lot of tiles everywhere, you won't actually be able to push through. So too much density is bad, but too little density is also bad. So you have to find that Goldilocks version in the Goldilocks range. Actually, give me one. Actually, something just came up. Can you give me 1 second? Sorry, hold up. I'll be right back. I had ordered food a little bit ago, and the guy decided to show up just now, so I sent my brother to go take care of that. Hello?
We're here. We're here.
Oh,
Go
okay.
ahead. Right.
We're here. We're waiting on you. So yeah.
Okay. Can you guys see the screen still?
You took it off presentation mode, but there you go. You're good.
Oh, okay.
You took it off. Now you're good.
All right. So basically, you would just want the Goldilocks range, and different gases have a different flow to the Pachet's curve. So the materials we use that I primarily focused on are in green, and so far it was just the Brockhorst objects, such as the E7400 flow controller system and the E7401 control system, and there was also an assortment of wires and what is it, in the 201 in the F201 what is it, flow meters. And very recently we received a glass tube and plastic shields that we were using to actually create the vacuum chamber. And the vacuum pump was a device that I actually was having difficulty with earlier. And I was working on that too. So that's going to be for future research. All right, so the E7401, this is basically just something we would use to calibrate the mass control units to control the amount of air that will be inside of one of the glass chamber tubes. Right now, this one was actually difficult to work with, and we actually didn't have all the materials for this one because we had the connections for this for the L1s through 4 plug inputs, but not the L5. So instead of working with those, we decided just to work with the E7400. So this next part is just going to be a what do you call it? It's basically going to be a step-by-step process as to how to get it to work. So right here, you would use these arrow keys to kind of push yourself through the menus and sub-menus. And let me see. So first, if you wanted to operate, you would go to the reset menu and kind of move around go to reset alarm, and then go all the way to the end. You have to input the password, which is ABC. And once you input the password, you'll have access to all the the counter menu, alarm menu, instrument menu, and local menu, which are really important. So then you would go to the operation menu and go to the control mode. And you would set it to keyboard and flow bus, And this will allow you to directly just input how you want the how much air and gas you want to go through the flow controller into the system. And then to the local menu, you would go to the display mode, which you would use to what do you call it? To display what information you want. In our case, we'd want the flow rate and the we want the flow rate and the set point. So what the set point tells us is how much air we're pushing through the flow meters, not the flow meters. And then the instrument menu, it would actually you would calibrate the you would actually put in the capacity, which is at 0% of the set point, which would be no air goes through, and then the sensor capacity, which would be how much, because each flow meter has a certain max range it can use, and you would just input that into that, and from there you would go to the measure menu, and you would edit the set point, and it'll allow you to actually put in how much gas you want going through. In our case, we're using normal liters a minute. So right here is an example of a flow controller, and we were using the Brockhorst high-tech ones. And right here, as you can see, these are like right here, it's a maximum flow is 6 liters a minute, and this is calibrated for nitrogen gas. And hold up. So typical setup, it would go from I would attach a wire from the E7400 into the flow controller, and the gas would go through here, through the cylinder and out the other end. Actually, no, wait, hold up, that's wrong. It actually go out through this way and then out the other. And I actually made that mistake earlier in semester. It actually cut me out 2 weeks of progress, but I managed to get it in the end. And so this was just the beginning to the design of the plasma tube. I wanted to actually paint this one black so you could see electricity better once we had all the parts installed. And this was just the setup for this. And this was the vacuum pump we were using. It was actually fairly new, and I had some difficulty with this one, but I was actually just getting on to the rascal, and I was actually just getting on the figuring this out so we could begin building the actual vacuum system. Not the vacuum system, the plasma generator. And so further work is actually going to be to actually complete the project, and hopefully I'll be able to finish that this summer. So we'll actually have a working plasma tube, because the equipment we've used has actually been sitting down there for a while. And I'm excited because since we got it to work, we'll actually have some use for it. And all the information from this slide was hyperphysics, just like some of the basic information on plasmas, and mostly from Brockhorst, because we were mostly using Brockhorst materials for this project. And that's it so far. Any questions?
Questions for David? So a few things that David didn't mention is that this particular design for a glow plasma is very similar to that of a fluorescent lamp, and it's a design that was developed in partnership with collaborators at the Princeton Plasma Physics Lab. They use it as a demonstration tool in their graduate program, and whenever they teach summer courses to professors. So this is what will be unique about our spin. What David has done to it is he's working with this flow meter, with this Brockhorst flow meter, and he's going to use, we're going to try different gases. The one at Princeton only works with air. So they bring it down to a vacuum, and then they introduce air. David's setup will introduce all sorts of gases, as he mentioned, using the Brockhorst reactor. And as you can see, it's not a trivial project, because programming any of these mass flow controllers is not straightforward. It's not the easiest of things, and especially you have to calibrate for the different gases that you're working with and all of these other different parameters. So this is an ongoing project, and the hope is that this particular setup that David is building here will be integrated into the advanced physics laboratory sequence that we have in Advanced Lab 2 or Advanced Lab 4, which will further introduce concepts of plasma physics. So the hope is that this will become not only a demonstration unit, but an actual lab that will be done by future students in the advanced physics lab. So let's thank David for his great job and his continued work, and move on to our next speaker. So David, good job.
Thank you.
Our next speaker is Erica Cairo. Erica is working with Professor Wang, and she is doing a project that is entitled, David, you have to stop sharing so Erica can share. And the project is entitled Dependence of Pdot, PSS, I'm probably saying this wrong, Erica will correct me, absorbance spectrum on spin coating settings.
Let me just share my screen. So, hi, my name is Erica Cairo, and my project is called Dependence on Pdot PSS Absorbance Spectrum on Spin Coating Settings. And as Dr. Lopez says, I am working with Dr. Wang. So in this project, we use the conducting polymer Pdot PSS, and conducting polymers can be applied to a very broad range of specialties such as solar cells, light-emitting diodes, sensors, and other optoelectronics. So in particular, the conducting polymer that we use, Pdot PSS, it's the most popular due to its high conductivity and stability, and it's been widely used as contact material in solar cells. So one of the application methods of conducting polymers is spin coating, and the different settings of spin coating, which can be the speed, the different layers that you put on the substrate, or the spin coating time, like how long you spin your substrate, can affect the thickness and absorbance of Pdot PSS, which may affect solar cell performance. So in this presentation, we show our studies on the absorbance spectrum of Pdot PSS with different spin coating settings. And the results of this research will help us determine the best spin coating setting for polymer applications on solar cells. Oh, sorry about that. So this is the different apparatus that I used in the lab. On the left, we have a spin coater, which is used to distribute the Pdot polymer as a thin layer on the glass substrate that you put inside. And then on the right, we have a UV-vis spectrophotometer, which is used to measure the percentage of light transmittance through the different samples that we made and we put inside the actual UV-vis. So these are the samples that we ended up making. There are two sets. The set on the the left, which is the first set, was made of only one layer of Pdot, and the set on the right, the second set, was made with 2 layers of Pdot. So when making the second set, you have to wait for the first layer to dry sufficiently, and then run it through the spin coater again, adding another layer of Pdot and letting that dry before you put it in the UV-vis. This is just a breakdown of how we made our samples. In each set, there are 3 samples, and each sample is spun at different speeds. So there's one spun at 1,000 RPM, 2,000 RPM, and 3,000 RPM. And each of the samples were spun for exactly 1 minute. So this is the data that we collected for the absorbance of 1 layer of Pdot PSS. As you can see, the range of light transmittance is very close together, and we deduce that this means that the data is inconclusive. And from the first set, it's inconclusive regarding which speed is optimal when using 1 layer Pdot on a glass substrate. And this is the data that we collected from the second set, and this is what we expected the data for both sets to look like. We expected that the slower the speed the substrate was spun, the thicker the layer of the Pdot would be on the glass, and the less light would be allowed through the glass, and which is what you can see when you observe the data on the graph to the right. So this is just all 6 samples put together on the same graph so that we can compare the 2 sets. So the data from the 1,000 RPM and the 2,000 RPM samples from the second set were as expected. The transmittance for those 2 samples were expected to be lower than all of the data from the first set, suggesting that they were thicker and less light was allowed through the glass. The one exception is the 3,000 RPM sample from the second set, and this was high. This had a higher light transmittance than all the samples from the first layer. So we speculate this is because as the second layer was added, the second layer dissolved some of the first layer, and the 3000 RPM speed was fast enough to spin off part of the first layer, meaning that the sample itself was thinner than all of the samples from the first set. This is just a closer look of the data all put together, and we can see down below how the samples are broken up. And so in conclusion, we found for the one layer samples, the range of the spin coating speeds 1000 to 3000 RPM didn't make much of a difference in the absorption spectrum. And for the two layer samples, the higher the spin coating speed, the thinner the film and the higher the transmittance, which was what we expected. And compared to the one layer sample, the two layer sample generally has a lower transmittance, which was as expected, except for that one exception, the 3000 RPM sample in the second set. It was thinner than expected, which might be due to the first layer being spun off as the second layer was added. So for our future works, this line of research will lead us to finding the combination of polymer layers and spin coating speeds that correspond to different thicknesses or different absorbances on the absorption spectrum. And we plan to try different spin coating speeds outside of the 1000 RPM range for the one layer samples to see if we can find a range that shows significant difference in the light transmittance. And we plan to try higher spin coating speeds for the two layer samples to observe if the transmittance can become any higher. And the next step is to calculate the thickness of the Pdot film using the absorption data. And for my acknowledge, I'd like to acknowledge my professor, Dr. Wang, who was my advisor through this project, and my colleagues, Anthony Sanaya Herrera and Brooke Richards.
Thank you, Erica. Great presentation. Can I ask you a quick question? Do you have any idea about the thickness of those layers?
So as of now, I don't know about the thickness of the layers because this was a very quick project. It did not take that long to put together.
One thing you can do is probably, you made a kind of a conclusion about the 3001. You said that we are speculating it might be thinner because it's eating up the bottom layer or something because of the fast speed. You can check the thickness. I don't know. We can try these in our ellipsometer, and maybe we can see that if what you said is really what's happening. Okay, in the ellipsometer, I don't know the thicknesses of these things, but I think these are measurable. I can see those Pdot layers, and if they have a different let's say index of refraction than the substrate, then we should be able to see the thicknesses. We can do that. Great job. Thank you.
Thank you.
As Alper mentioned, you can measure using the ellipsometer, and then we can also calculate it using the conductivity measurement. So that's in her future work. Yeah.
Very good. Very good.
Yeah. Yeah, and Erica is a freshman, so this is her work during the COVID time. Yep. So
yeah. Good job. Good job.
Good job, Erica.
Thank you.
So Erica still has plenty of time to get a few publications, right, waiting out of your lab.
Yes, hopefully when this is all over, she can stay in the lab a lot more time.
Great. Thank you, Erica. Virtual applause to her for a great job done here. So our next speaker is Brooke Richards, and Brooke Richards is further working with Professor Wei-Ning Wang, and her presentation is entitled Studies of Cadmium Telluride, I can never say that right, and cadmium sulfide solar cells. You seem to be in the lab. This is promising.
I figured this is the safest place so that no one would barge in here and be obnoxious while I'm trying to present. All right. So, like Dr. Lopez said, my name is Brooke Richards. I'm a sophomore in the dual degree engineering program, and I'm going to talk about a project that Dr. Wang and I have been working on for the past couple of months, and that is the simulation on CDT-CDS solar cells. So first, I'm going to talk about the CDT-CDS solar cells themselves, and then I'm going to explain the software that we use to simulate these cells. And then I'm going to explain the results we got from the simulation runs. And then I'm going to compare our results to the results of a different solar cell simulation software. And then I will talk about the next steps of this project. So one of the most studied thin film solar cells are the cadmium telluride, cadmium sulfide solar cells. And then over the years, their efficiencies have reached about 22%. However, we want to increase that to make the quality of the solar cell better. So in order to do this, we have to understand how the efficiency depends on the different parameters of the solar cell itself, such as the thickness and the doping levels of each layer. So in order to do this, we use a simulation software, and that did successfully show us how the efficiency, along with other parameters of the solar cell, depend on the thickness and doping level of each layer. And then to the right, I have a picture of or a diagram, I should say, of a CDT-CDS solar cell. And the transparent conductive oxide layer that we use in our lab is the tin oxide, or I should say for this study is the tin oxide, which is SNO2. So our software name is called WX-AMPS, and this was created by some professors at UI, UC, and PSU. And then to the left, you can see that we can set the temperature and the type of light we want to shine onto the solar cell for our runs. And then the middle picture shows the number of layers you can put on your cell and the thickness, the amount of thickness you want to put onto them. And then also to the right of that picture, you can see how you can adjust the other properties of the cell, such as the band gap, the affinity, and the doping level of each layer. And then to the right, you can see the IV curve that is produced. And then also to the upper right of that picture, you can see that it also shows you the VOC, which is the open circuit voltage, which is the maximum voltage available from a solar cell to use. And then the JSC, which is the short circuit current, which is the current through the solar cell when the voltage is equal to 0. And then the FF is the fill factor, and that's the ratio of the maximum power of the solar cell to the product of the VOC and JSC. And then the one we focus on the most is the efficiency, and that's basically the amount of sunlight that gets to the solar cell that eventually turns into electricity that we can use. So the first thing we changed was the thickness of the CDTE layer. And if you look to the upper left picture, that's the efficiency graph. And you can see that the efficiency increases as the thickness increases, but only till about 2.5 to 3 micrometers, and then drastically decreases after that. And then the VOC does increase as the thickness increases. However, the fill factor and the JSC do decrease as the thickness increases. Next, we change the CDS thickness. And if you look at the efficiency graph again, it's the opposite of the CDTE layer. The efficiency does decrease as the thickness increases. And this is the same for the other parameters. The VOC, fill factor, and JSC do decrease as the thickness increases. And then we looked at the dependence on the tin oxide layer. And it's kind of similar to the CDS layer, where all the parameters we look at here decrease as the thickness increases. But if you look at the range, especially on the efficiency graph, the efficiency percentage only decreases by less than 1% as we increase the tin oxide layer from 0 to 1 micrometer. So it decreases as the thickness increases, but not as much as the other two layers, as the CDS layer. So after we looked at the thicknesses of each layer, we looked at the doping levels of the CDTE layer. And if you look at the efficiency graph again, you can see that it levels off and reaches a stable level of about 20% once the doping level reaches around 2 times 10 to the 15th per cubic centimeter. And then it only slightly increases as you increase the doping level after that point. And then we looked at the doping level of the CDS layer, and the efficiency reaches a stable level of around 18.5% once the doping level reaches around 1.1 times 10 to the 19 per cubic centimeter. And similar to the CDTE doping level, the efficiency only slightly increases after you increase the doping level at that point. So once we got our own results using the WX-AMP simulator, we wanted to compare them to the results that use a different software. So we found a study that also observed the effects that layer thickness and doping levels had on the solar cell. And that project used SCAPS, which is Solar Cell Capacitance Simulator, and that was developed at University of Ghent. And you can see to the left, I have a snapshot of their software. And then to the right, I have the JV curves that they use to show their results. So once we finished comparing the results with the WX AMP results with the SCAPS results, we saw that in general, our trends were very similar, especially concerning the efficiency percentage of the CDT, CDS solar cells. For example, concerning the thickness of the CDS layer, with both simulations, as the thickness increased, the efficiency decreased. And then also with the thickness of the CDTE layer, the maximum efficiency was reached around 2.5, 3 micrometers for both simulators. So to summarize our results, we found that the maximum efficiency was reached when the CDT thickness was around 3 micrometers, the CDS thickness was about 0.025 micrometers, and then when the tin oxide thickness was around 0.05 micrometers. Also, we saw that the efficiency reached steady levels when the doping level of the CDTE layer was around 2 times 10 to the 15th per cubic centimeter, and when the doping level of the CDS layer was around 1.1 times 10 to the 19 per cubic centimeter. So the next step of this project is to simulate a CDTE polymer solar cell that's here in the lab, and then compare those simulations to simulation results to experimental results. And some people I'd like to thank for their help with this project are first my advisor, Dr. Wong, for all her help and letting me work in her lab and teaching me how to use the software and everything. And then my other 2 colleagues, Anthony and Erica. And then I also like to thank the physics department and then the professors at UI, UC, and PSU that developed the SCAPS software. And thank you for your time. Does anyone have any questions?
Thank you, Brooke. Great presentation. Thank you.
Questions? Okay, so a sign of a good talk is you left everyone silenced. So thank you, Brooke. And Brooke, you're a sophomore this year, right?
Yes.
So Brooke has 2 more years, so Dr. Wong should get 2 or 3 publications out of your work.
No
both you and
pressure,
Eric are the
Brooke.
future of the work that's going on there. So keep up the good work.
Thank you.
All right. Thank you, Brooke. So our next speaker is, and I love his name. I love all Spanish names because they just keep adding names and names. So Anthony Sanani Herrera. And Anthony is also working with Professor Wong. As you see, she's doing a great job with our physics majors. And Anthony will present a research talk entitled Study of Thickness Dependence on Tooling Factor. So Anthony, Anthony Sanani Herrera.
Yes, just give me 1 second.
Yep, go ahead.
Let's see if this works. Is everyone able to see the presentation?
Yep, it's working, Anthony.
Okay. As was introduced, my name is Anthony Sanai, and my advisor is Dr. Wang. And today I'll be presenting my research on thin film thickness dependency on tooling factor. So just a quick introduction on what thin films are. So thin films is basically a layered material that's fraction of a nanometers in width. We use them in solar cell fabrication to create electrodes, and they're also used to have other applications outside of solar cells. The thickness of the film is very important as the property can change as the thickness increases. For this reason, most deposition machines have sensors that monitor the thickness of the sample. And a problem that we encountered when using our evaporation deposition machine was that the reported thickness was off. So we set out to find out what could be causing this. And so as you can see from the left-hand side, this photo was taken in fall of 2020. So at 500 angstrom deposition, you can see how the vacuum chamber has a layer of deposited material, a clear layer. And then if you look onto the right picture, you can see that the same deposition monitor with this giving out the same 500 angstrom deposition thickness, there's almost no layer of deposited material there. So what could be causing this? First of all, I would give a sort of slight overview of what the deposition machine is and how it works. So for the evaporation deposition to work, a vacuum must exist in order to get the material to deposit onto the substrate, which is where the sample is. We use pumps, a series of pumps to create this high vacuum in the vacuum chamber. The boat, as you can see where it's labeled source material, it is used to hold the material that is being deposited and also to conduct current through it, which is used to vaporize the material and have it evaporate. The rate of deposition is controlled by controlling the amount of current that's passing through it. So the more current you put through it, the more material will be vaporized and the more material will be deposited per second. And the materials we use for deposition range from gold, copper, aluminum, and each material has its own requirements for deposition. But for our research, we use aluminum as its vacuum requirements are a lot less extreme. And also it's a lot cheaper to do multiple runs with aluminum than it is with something like gold. And then there's also as part of the system, there's the Infocom deposition monitor, which is a quartz crystal monitor that uses quartz crystals to measure the amount of material that's been deposited by measuring the frequency change and relating it to the amount of material deposited onto the crystal. This crystal is mounted inside the vacuum chamber above the source of the evaporation. And this device is what we use to read out the thickness that's supposed to be on the substrate. So our first point to check was there for any possible error. The QCM, the quartz crystal monitor, has this thing called tooling factor, which is pretty much a measurement of the ratio between the amount of material that's been deposited onto the crystal and the amount of material that's been deposited onto the substrate. The tooling factor depends on the position of the sensor when compared to the substrate. As you can see from the picture on the right, if the monitor is above the substrate, we should use a tooling factor over 100% because there's going to be more material deposited onto the substrate than that is being deposited onto the quartz crystal. Whereas if the sensor is below the substrate, then we would need to have a tooling factor under 100% because there's more material being deposited onto the quartz crystal than onto the substrate. So then how do we find the tooling factor? We know from the last slide that the tooling factor has to be below 100, but how are we supposed to find the actual thickness of that's being deposited if what we use to monitor the deposition is not giving us the correct thickness? For that reason, we'll do trials testing out different tooling factors and using a machine called the UV-Vis to get the thickness of the material by testing its light absorption. So the UV-Vis thickness measurements. So it uses light. The UV-Vis uses lights across the spectrum to be able to test the absorption of the sample. Using research, we found that sort of gives a correlation between thickness and the transmittance of the sample. We'll be able to compare the theoretical thickness to the
I don't know.
Yeah, it looks like we lost Anthony. His
Yeah.
Wi-Fi might have, or his laptop.
Yes,
I've had a few students. There we go. He's back.
All right.
I'm sorry. I completely didn't realize that my computer ran out of battery. It was running out of battery during the other presentations.
Okay.
Okay. Are you able to show your talk, or you just want to get to your conclusions, or?
I might be able to share the PowerPoint. Just give me 1 second. There we go. Yeah. Okay.
Yep. What's up?
There we are. I'll just quickly go back to where. Yes. So in order to get an accurate measurement of what is being deposited onto the onto our sample, we need to be able to factor out the absorption of light that's coming from the sample we're using, which is the glass slide in this case. So we use that as a baseline, and then afterwards, as shown in the picture below, we put it into the UV vis chamber, and we test for the transmittance through the glass and the aluminum that's been deposited. And with the baseline, we're able to factor that out and then get the actual thickness of the aluminum. So our results from this, using the UV vis, we found that this was significantly off. As shown in the top right, this shows the at 300 nanometers what the Armstrong thickness is and what its transmittance should be. And as shown from in the results, at 300 nanometers, there is a significantly larger transmittance, which in turn leads to it having actually deposited a thinner layer of material than has been reported. So for a 90 tooling factor, the estimated percent T should be 0.1, but from our measurements, the average of the two slides that we used turned out to be 2.4. And then for 85, for a tooling factor of 85, the actual percent transmittance was 0.9. So it shows that the original tooling factor that was set up for the QCM is significantly off, and it's causing a thinner layer to be deposited when a thicker layer is being reported by the QCM. So in conclusion, I have shown that by changing the tooling factor, the measurement of the thickness monitor could be corrected. Also, that the tooling factor needs to be calibrated more precisely in order to provide an accurate measurement of the material that's being deposited. And that I also seems to see that the further the center is from the substrate, the more drastic the correction is. For future work, I think that more depositions will definitely need to be required to pinpoint the tooling factor value that gives the most accurate readings for our current setup, and also that we should work with other materials in order to ensure the tooling factor is not something that's specific to different materials. And I'd like to acknowledge Dr. Wong, my advisor, who's taught me how to use the deposition machine and the UV vis, as well as Erica, Brooke, and the entire physics departments for helping me along the way. Yeah.
Thank you, Anthony. Thank you very much.
All right. Thank you, Anthony. Any questions for Anthony? I like how, Anthony, you were able to recover even after your computer died. So good job doing that. So this ends the research presentation part of our event tonight. So I'd like to thank all our speakers, starting with Will earlier, with Will Davis and his master's thesis presentation, and everyone and every other student that has presented their presentation. In particular, we'd like to thank, and maybe that you can turn on your cameras, maybe Anthony's not able to, but our graduating seniors. So Jasmine Emerson, who was graduating this year. David Turner, who's also graduating this year. Ramiz, I believe you're graduating, right, Ramiz? I haven't seen you in so long.
I am graduating.
Yeah, okay. I figured you were. And also graduating Alexis. Alexis, as we all know, is one of the hardest working people in physics. So Alexis is graduating, and am I forgetting anyone? And Anthony, I said, right? So I believe that is everyone who's graduating. So let's give them a round of applause for all our graduating seniors and all the work that they did over the next few years.
Thank you, guys. Thank you.
Thank you. So for now, for our final part of our program tonight, we will have the Dr. Seton Kim, PhD Medal for Excellence in Physics. And this is the award ceremony to give the Kim Medal. And I don't know if Dr. Sahiner would like to say a few words, and then Dr. Troja will give a presentation on who Dr. Kim was. And then we will award the Kim Medal.
Okay, we are getting a little late because of the presentations, but so I'm not going to make an introduction, but I will leave to Dr. Troja to the floor. And this is our once a year award for the graduating senior for the achievement of Dr. Kim was a physics professor for years, and Dr. Troja, with Dr. Troja's help, we set up this one, and he's going to talk about Dr. Kim's achievements, and then we will present the Kim Medal to this year's recipient. Thank you. Dr. Troja, go ahead.
All right, so we'd like to go and introduce some of the background on the Kim Medal and ultimately what fostered our ideas for introducing that. Okay. So Dr. Kim taught physics here in the department for 42 years, though, and he was a great inspiration for all of us, not just for the students, but also his fellow faculty members, though. So where did the idea of the Kim Medal come from originally? Well, historically, starting back in 1861, Seton Hall College decided to formulate a bunch of different medals that would be awarded for various things. The very first one was the Hamilton A. Hearn Gold and Silver Medals for good conduct. They then went and added a number of different academic medals, and these were all backed up with various small monetary prizes, though. At one time, they ended up having a total of 36 medals that were given on out. Okay. And the one that we're interested in was the so-called Berry Prize. All right. Patrick Berry, he was, along with his business partner, the owner of a global nursery, the very first global nursery. Back in the 1840s, they were shipping exotic plants from all over the world. At that time period, it was absolutely unheard of, though, and a lot of people who were well off wanted to get their hands on exotic plants to put into the private garden stuff. So they became very wealthy overnight. His son, William Berry, was a student at Seton Hall College, though, and so he got to know President McQuaid back then, and it became very good friends, and so he started sponsoring this medal. Originally, it was given out for the natural sciences, and then later on, chemistry got their own medal, so it became the Berry Prize for Physics, though. He continued sponsoring it until he passed away in 1890. The business went into the hands of his son, who also continued to sponsor the medal until his untimely death back in 1916. After that time period, though, all the other medals just disappeared. And so the time period when they handed out medals ended. Or it was not until we introduced the Kim medal that finally medals started to be given to students here at Seton Hall. Okay. So we want to understand about Dr. Kim, why he was so significant. Right. So to go back to his beloved native Korea, his grandfather, who was Kim Ka-jin, was influential in the government back at the time period, though. His grandfather was a member of what they call the Independence Club. Korea was hoping to transition from having a monarchy to a democracy, and so they were working to try to improve things. His grandfather was a minister of culture, commerce, and public works, and he also held different government positions, including he was an ambassador to Japan for a while, and he was a special envoy to the Chinese emperor. All right. Korea had been a tribute state of China for centuries. All right. And so they kept on having to send monetary contributions to the emperor. Well, they decided they didn't want to have that anymore, though, so they destroyed the symbolic tribute gate that was set up there, and they created this. This is the Independence Gate that they had later on, though. And so even to this day, though, people know about Kim Ka-jin and his contributions to society. Unfortunately, in 1910, Japan decided to annex Korea. It was a very brutal colonization that took place, though. Kim Ka-jin had to flee along with his eldest son and his family to China in 1919. He was effectively the government in exile until his death in 1927, though. Dr. Kim's father, Kim Kakan, was unfortunately not able to escape, and him and several of his siblings had to live underneath the Japanese occupation, and it was into this environment that Dr. Kim was born. Dr. Kim's father was a high school teacher and a university professor of English, and he instilled in Dr. Kim importance of education, as well as the dedication to public service that came ultimately from his grandfather. Right? Finally, 1945 rolls on around, end of World War II, the Japanese are defeated, and the people of Korea feel that, oh, finally we have our freedom back and we can go and start rebuilding. That was unfortunately short-lived, all right? In 1951, the North Koreans invaded South Korea. Dr. Kim was separated from his parents. He later learned that they were unfortunately killed in the invasion, though. One of his uncles was basically taken prison by the North Koreans and never heard from again. Dr. Kim's father had taught him English. He was able to go down to the port area of Busan. That's where the Allied troops had amassed there, trying to push back the North Koreans, and he encountered the men from the 204th Field Artillery Battalion, which is made out of people from the Utah National Guard. All right, and because he could speak English, they allowed him to work in the mess hall there, and he also acted as an interpreter. Right after the situation is stabilized a little bit, the men of the 204th Field Artillery Battalion went back home, but Dr. Kim kept in contact with them. Right? And he wrote to Colonel Whitesides, telling him about his dream of coming to America to obtain an education. He was so enamored with that idea. And in fact, at a very early age, he had already conceived of the fact that he wanted to get his PhD. So Colonel Whiteside said, we have to help this young man, though, and they put the word out, not only to all the men that were in the field artillery battalions, but also to the local populace, though. And there was articles in the local newspaper, though. They had fundraisers to raise money to be able to pay for his transportation and everything. Someone donated an apartment he could stay in while he was attending college, gratis, all right? Other people donated clothing and everything. So they had this whole entire effort to bring him to the United States. And that actually came to pass. So in December of 1954, Dr. Kim arrived here in the United States. And not only was this heralded in the local newspapers out in Utah, but literally it made national news. Newspapers from all across the country carried the story of how these Utah National Guardmen raised the funds in order to bring Dr. Kim here to the United States for his education. And he did not disappoint. He went and attended the Utah State Agricultural College, which is now Utah State University. He did very, very well in his studies there. All right, became, of course, parts of several different clubs here, including the Cosmopolitan Club here, which is formed by international students. And of course, he was very prominent in the physics club. He did so well there. He went and was able to apply and be accepted by the University of California at Berkeley's Department of Physics for PhD studies out there. While he was out there, he performed his research in experimental and theoretical nuclear physics at Lawrence Berkeley Laboratory, which is up on the hill above the UC Berkeley campus. His research went on to be published in prestigious journals like Physical Review, for example, and also there were presentations made around the world based on this research, including over in Russia, which during the Cold War it was almost unheard of. Okay. After he received his PhD in 1965, he had a postdoc position out in Illinois. And then, as fate would have it, Dr. Eugene Petrick, who was the first department chair here in the Department of Physics at Seton Hall University, decided to leave this position and become a vice president of a college out in California. That provided an opening, and Dr. Kim applied for that, and he was hired. And over the next 42 years, he taught thousands of Seton Hall students, though. For those of you who have taken classes in the introductory physics laboratory as well as the advanced physics laboratory, though, Dr. Kim was the one that pioneered many of those experiments that we perform in those classes, though. He was always a well thought of teacher. When he went out through the years, the students always really enjoyed listening to him. And when we had his retirement party in 2011, President Esteban stated at the time period that he had estimated that Dr. Kim had educated over 12,000 students during his time period here at Seton Hall. He always emphasized rigor in the science and, having great dedication to studies of physics, though. So when we decided to develop this medal, you have certain students who are very good academically, others are very good in the laboratory, and still others are intertwined with SPS, for example, though. But it's the rare student who is good academically, is going to go and promote physics both here at Seton Hall and beyond, and has excellent research capabilities, though. So from a vote of the faculty here, we've decided on the student who emphasizes all three of those subsets that lead to the best of the best for our students. Usually, before we had unfortunately put up with the pandemic, though, the Kim family would come and announce the winner of the medal. I did talk to his daughter Kathleen last night, though, so she's very excited about this, though, but unfortunately could not be with us today. And so I'm going to go and turn this over to Dr. Shahiner, and he will announce the recipient for this year that will join all these illustrious past recipients.
Thank you, Dr. Tura. Thank you for excellent presentation. Okay, as Dr. Tura says, usually the family, Kim family awards that, but Catherine couldn't be able to do that. So I will announce, and there was a letter coming from the Vice President of the Advancement of Seton Hall, and I'll read that and I will present the award. And in addition to the Kim medal and the $1,000 to the recipient, there's also a matching funds for physics department from the Pfizer company with Dr. Catherine Kim's company for the research. And so we've been receiving those funds from the Pfizer company too. Okay, let me read the letter from the John Paparesnos from the University Advancement, and here it is. Our awardee Kim medal recipient is Jasmine Emerson this year. Congratulations, Jasmine, first of all. Can I see where Jasmine is? Okay, this is the letter. Dear Mrs. Emerson, I'm pleased to inform that you have been chosen as the 2021 recipient of the Seton Kim Medal for Academic Achievement in Physics. While many students were considered, the Department of Physics deemed you the most deserving candidate. The medal is named for the late Dr. Seton Kim, a beloved professor of physics at Seton Hall University who devoted his academic life to education of students in the sciences. The medal is awarded to a student who has demonstrated excellence in the classroom while promoting the ideals of physics both at Seton Hall and beyond. It is also a recognition of excellent research. Department of Physics has bestowed on you this high honor of recognition in of the academic achievements you demonstrated during your tenure at Seton Hall University. The Department of Physics is pleased to bestow upon you this medal, which is accompanied by a financial award of $1,000 made possible by the generosity of the Kim family. On the behalf of the entire Seton Hall community, I congratulate you on this most prestigious award. I wish you continued success in your academic pursuits. That's from the advancement present, and from my heart, you deserve, Jasmine. You're the this year's Seton Kim Medal recipient. Congratulations, and I'm sure that you're going to be promoting physics and physics department and materials for the rest of your career. Thank you.
Thank you so much, Dr. Seton.
Let's have a hand of applause for Jasmine, our Kim Medal, and then I'll leave the word to Dr. Lopez to close the event.
Oh, congratulations.
Thank you. Thank you, Jasmine, for your great work.
Thank you.
Jose, you're muted.
Congratulations, Jasmine. I don't know if you'd like to say a few words or something.
I just want to say thank you so much. I mean, the physics department here has been absolutely amazing. I remember my freshman year, of course, like all the other physics students, our very first physics professor is Dr. Sahiner, and I was immediately very interested in the research that he does, and him and the rest of his research lab welcomed me with open arms. And then just going further and working with Dr. Troha and Josh, who's also attending with the general physics students and helping with their recitations, and then the principal physics students and helping them just do well in their courses.
it's been a great opportunity, and I want to thank you all so much.
Thank you.
Congratulations.
Congratulations, Jasmine. Thank you very much. So I'd like to thank all our participants tonight. Congratulations to Jasmine for being the 2021 Kim medal winner. So you will get a physical medal also at one point, hopefully for graduation, so you can display it. And we'd like to thank all our graduating seniors once again, as I mentioned earlier, David Turner, Ramiz, Al-Gilat, I'll get it one of these days, Ramiz, and also Alexis Duffy. Congratulations to all our graduating seniors, and congratulations to all our presenters this evening. And I would also like to thank, as I mentioned earlier, the members of the organizing committee of the Petersheim, which include Professor Wong, who is the secretary of the Petersheim organizing committee, Dr. Suli Chang, who is the co-chair of the longtime serving chair of the committee, and also Professor Schroen from the Department of Physics, not environmental studies, as it says in her description. And I noticed that Mike over in environmental is your manager. That's not true. It's Alper is your chair. So we have to make sure to fix that. And
glad to have Martha all the time.
We're trying to take Martha away. And then we'd also like to thank Professor Lisa Rose Wiles from the University Libraries, who is our reference librarian, and you should use her in case you're doing research. She is a treasure at the university for that. So we'd like to thank all members of the Department of Physics. So I thanked Dr. Wong already. Professor Mitra Fazabadi was here earlier. Professor Kalani, who left, was also here earlier. And of course, Professor Troha. And without forgetting, of course, Professor Alper Sahiner, who is our department chair. So thank you, everyone, for participating with us tonight. And we look forward to seeing more incredible things in the near future. Congratulations.
Thank you, Dr. Lopez, for organizing this wonderful event. Thank you. Thank you all.
Thank you, everyone. Have a good night.
Bye-bye. Bye-bye.
Good night.
Night.
Good night, everyone.
Good night, everybody.
Good night. Congratulations, Jasmine.
Thank you.
Good night. Thank you.
Thank you. Thanks, Brooke.